Well, hi everyone. Welcome to the 14th EveryLife Foundation Scientific Workshop. We are so thrilled that you all can join us here today, and welcome to those who are watching us on the live stream. The scientific workshop really is focused on advancing the conversation on topics that sit at the intersection of science and regulatory and legislative policy. And today's topic is the perfect example of such a topic, where the advances in science may be limited by policy barriers. So first, before we get started, some housekeeping tips and thank yous. Hopefully, nope. We'll get there. Okay, so first, thank you to all of our sponsors, many of whom are here today, for helping make this workshop possible and making it possible to convene all of you, to have wonderful patient advocacy group participation, and just the discussions we have today. And thank you to our planning committee. We have many esteemed members of our planning committee you see here, and we'll also send a big thank you to one additional member of our team, Bailey McGowan, who's not on here out of a desire to keep the symmetry of the slide. Her own doing, by the way, but very grateful for Bailey's support as well. Yes. A couple other housekeeping before we jump into the polls that you're going to see here. Is it going to pop up? Okay. So couple housekeeping tips. We have speaker bios on your tables. If you want to read our speaker bios, because we are going to save time by not going in depth today, because we could go on about all of them, you can scan the QR code on your table to learn about our speakers. We are live streaming today, so please, when we get to discussion periods, make sure that we have mics when you are making comments or asking questions. And throughout the day, we will have a few live stream polling questions for you to complete. And we're also going to share a little bit about the survey that we conducted with our patient advocacy groups that align with the survey questions you're going to fill out here today. So to get us started, and I'm sorry, one more, thank you, Bailey. There are baskets on your table. We really want some robust discussion throughout the day, but we know that sometimes you have comments you want to share offline, or we may not have time to get to everyone's comments. So please utilize your notepads and the baskets on the table to leave us your comments and insights that you want to make sure are incorporated into the summary and output of today's discussion. Okay, so to kick us off, we want to ask two quick polling questions to get a pulse of the room. To do that, okay. Oh, I see you scan the QR code. There we go. Sorry, scan the QR code if you're able to with your phones, and that should bring up a link right to Polls Everywhere. pollev.com/EveryLife Foundation 230. And if we can't get it off this, we'll make sure in the later questions we have we pass around the QR code. It is pollev.com/EveryLife Foundation 230, or you can text EveryLife Foundation 230 to 22333. We'll give everyone just a minute. And the first question is, what does ultra-rare mean to you in 5 words or less? If you are putting more than one word, please make sure you separate each word by quotations and underscore. Extremely small. Give people 30 more seconds to get in. seeing some common themes that are shared with our patient advocacy surveys. You'll see in just a moment. Unmet needs, very small challenges, but patience first and foremost. Wonderful. All right. Well, we'll move on to our next one. As you can, continue to share with us again, what does ultra-rare mean to you? That's why we're all here today. So the next question, the law, as all of you know, defines a rare disease as one that affects less than 200,000. Similarly, as we're here to discuss, do you believe a formal definition for ultra-rare diseases should be created? Okay. Great. All right. Well, I think you see the general pulse of the room. We have a little over 60% who feel yes solidly, 4% with no, and then the remainder is off screen, but it is unsure. And I think that really parallels quite closely the results that you saw that you will see in our patient advocacy group survey. So I wanted to share before I turn it over to Tim, who is going to be our official moderator for the day. I wanted to share just a few quick insights from that survey, and then you'll hear some other snippets throughout the day. And so we also asked our patient advocacy groups in Community Congress. We had about 78 responses within the last week alone. So thank you, especially to all of you online and who are in the room who filled out the survey. We asked you all to share a little bit about your thoughts around ultra-rare challenges. You're gonna again hear more throughout the day. But first and foremost, what do patient advocacy groups think should be done about defining ultra-rare formally? And I think it mirrors the pulse of the room a little bit more on the yes side, but there's definitely a solid group of responses that were unsure as well. And then, what does ultra-rare mean to patient advocacy groups? And you see the similar word cloud. The overwhelming, themes that dominated our responses were that, extremely small. Everyone could agree, that it means extremely small. But then from there, the numbers varied over the span of maybe 100, less than 100, all the way through to some people saying, more around 20,000, 50,000. Some people provided some incidences. And this really aligns with the very varied use of the term ultra-rare, both formally and informally, by regulatory agencies across the world, as well as in foundation work, in health technology assessment work, and across our own references and how we refer to ultra-rare. So as you can see, there's quite a lot of variability, but ultimately why we're here are these quotes coming from patient groups and what it means for them to be an ultra-rare patient organization. And I think these are just some highlights to kick us off, really ground us in why we're here today. And as we move forward, and I introduce Tim to start our formal program and cover our workshop goals, I just want you to keep in mind that patient perspective, and no matter how we talk throughout the day, it all comes down to how can we help make sure that we're advancing solutions for patients at the center of our conversations. So with that, I want to make sure, introduce Tim Franson, who is our moderator for the day. Tim is currently the principal of Fager Drinker Consulting in the firm's life sciences regulatory consulting practice, but Tim spent a large part of his career with Eli Lilly & Co., and he retired in the leadership role of Global Vice President for Regulatory Affairs. But most importantly, all of us in the patient advocacy community know Tim really as a skilled facilitator, a consensus builder, and a regulatory counsel to many patient advocacy groups across the rare disease space. So we're really thrilled and honored to have Tim help us moderate the discussion today and ensure that at the end of the day, we can reach these goals with all of your input. So thank you again to our sponsors, our planning committee, and with that, I'll hand it over to Tim.
Thank you, Jen. Well, thanks, Jamie, and so good to see so many folks engaged here and willing to come together to talk about a very complex process. So I have the pleasure of introducing our speakers, and one of the things we'll start with operationally is just to ask folks to introduce themselves. It's an opportunity for relating directly. You don't have someone secondhand like me talking about the accomplishments that probably mean much more to you to be able to share with your colleagues. So our topic today is incredibly interesting and challenging for a number of reasons. It's important to talk about how big this group is, and you've seen in the queries that there are differences in what one would think of in terms of quantitatively as ultra-rare. On the other hand, this is not just a game of numbers. It's defining a particular group such that if they were properly constituted and defined, so what? What would that mean? What could be done? And are there particular levers that one could operate to help more advantage those communities? So those kinds of levers might be various things that we see in other clinical trial and care environments. And to give you a kind of anchor on how we might hear from many different perspectives, and that is the goal today, to assure that those with diverging voices and views on these kind of topics have the opportunity to share and listen to others about perspectives from an industry side, from a regulator side, from patient advocacy, from parents, and to then be able to, as we go through the remainder of the day, begin to integrate some of those thoughts and arrive at whatever consensus comes through as a result of the discussions. So to give you an anchor for that, I'd like you to think of the acronym that comes from the word CARE, because we're talking about patient care in one way or another in everything we're doing. Think of CARE as an acronym for at least 4 of the major levers that might be pulled as when we talk about ultra-rare populations. So C for clinical, both clinical care and clinical development and the complexities therein. A for access to care as well as access to trials. R for regulations. Do we have the proper kind of incentives? Are incentives appropriate or not? And what kind of incentives might those be? Not just regulatory, but in other ways that would stimulate research. And then E for ethics. What are our obligations in a moral and ethical sense? And I believe most of our panels are designed to try and give airing to all those particular viewpoints. So as you see in the construct of the program, we attempt to invite each constituency to be able to share. And just a reflection as we go into this in terms of progress. When I started medical school last century, life was different. Life was different for all of you too, I'm sure. I see many youngsters here who say, last century, that's way long ago. But rare diseases were almost an afterthought. There was no such thing as patient-focused drug development. There was not an opportunity to really engage in the proper diagnosis of many of these things. Genetics was more of a theoretical than a practical discipline in those days. So to be able to reflect on not our frustrations now, because we all are frustrated by how less than rapidly things move for the well-being of patients, but understanding that over the last several decades, what we have seen built upon a foundation of patient-focused drug development and other similar design issues have allowed us to really begin to advance further. And we probably wouldn't be having this discussion today without those things. So I think it's important to celebrate what has been brought forward and then build atop of that. And as we talk about ultra-rare, assuring that we look at that as a distinctive way, if we can sense that in that fashion, to be able to leverage something that is advantageous in care, in clinical, access, regulatory, and ethical bounds for those with whom we work for and with in the patient community. So without further ado, I'm going to begin to bring our speakers forward. And I ask you all to give each of them proper and respectful attention, and also to think of proper and challenging comments that you'd like to bring forward, because there will be opportunities for those from the floor to speak. So our first three speakers, and they will come up sequentially. First, Sharon King's going to talk about patient advocacy and group perspective. Second, Adora Nadeau is unable to join us, so Amanda Malikoff is going to present on her behalf. Amanda will come up second to talk about the industry perspective for ultra-rare. And then third, Dr. Janet Maynard from FDA, the Director of the Office of Rare Diseases, will be talking about the U.S. regulatory perspective in this regard. After we then hear each of their comments, we will bring forward them plus additional panel members, and this will be chaired by Ethan Perlstein. And we will be talking then about considerations of small populations and what in total may be done. So that gives you a flavor of where we're headed here. And without further ado, Sharon, would you like to come forward, introduce yourself, and regale us with your experience?
Good morning, everyone. I can introduce myself pretty simply. I am a rare disease advocate, and I have been a rare disease advocate since July 24th of 2006. That's the day my daughter Taylor was diagnosed with a rare disease, very small population. And it's the day that I decided things had to change. And I'm going to tell you why. First off, I have a couple of slides. Thank you. And I also want to thank the EveryLife Foundation for inviting me to be a part of this important discussion. When you open the Michael J. Fox Foundation for Parkinson's Research website, this is the first thing you see on the screen. And I really like this quote, and I wish I'd been the first one to say it. You see, I believed in its relevance for many years. I remember the day informed optimism became my truth. It was a beautiful, sun-drenched morning, and I was on the phone with a dad who had 2 children with CLN2 Batten disease. We discussed our shared challenges as we tried to do our part in moving forward drug development, each of us for a form of Batten disease. And he asked, Sharon, how do you keep going knowing that the time and effort that you're putting into, trying to drive forward research for a therapy, a potential therapy for Taylor and other children like her, how do you keep going knowing that it's not going to be in time for her? And, so the conversation was all about the hurdles of drug development, but looking back, it was more about the loss of time with our children. Anyone who understands progressive illness knows that time is almost as much the enemy as the disease itself. Not only were we losing our children, we were losing precious time that we had left with our children. But our justification was that stopping would be a loss of hope for us, a loss of hope for our children, and a loss of hope for all the children who would follow ours. We had to keep going. People who know me well also have heard me say time and time again that hope is an action word. Hope is also informed optimism. There was a point along the way when I knew that the work Taylor's Tale was doing wasn't going to benefit Taylor. And I think that knowledge came pretty quickly. But after it did, I was no less committed to my goal, which was the potential for a better life for my child and all who would follow. While hope looked different for me at different stages of Taylor's illness, I was only hopeless, I can promise you, I was only hopeless for a matter of hours right after her diagnosis when we heard there is no treatment for CLN1 disease, and it's doubtful that a treatment will be developed in your daughter's lifetime and most likely in your own lifetime. It's just too small. Take your child home and make happy memories while you can. We heard those words. How do you make happy memories when you've just received that kind of diagnosis? That is hopeless. And I knew within 24 hours that it had to change. I could not and would not live with that kind of hopelessness for any length of time. And so Taylor's Tale was born to bring light to that tunnel of darkness. Optimism ran high during the proof of concept phase of an AAV gene therapy that Taylor's Tale funded for CLN1 disease beginning in 2013. When our treated mice celebrated their first birthday, we threw a big birthday party because it was a big moment in time. Mice with CLN1 disease don't often live far past 8 months of age. And at the end of the 3-year study, the data was good enough for us to find a new home for the project. Industry took on the research, and 2 years later, the program received clearance for an IND with a novel dual delivery. This, again, was another big deal for this patient community, this very small patient community with no available treatment options. You could see, feel, and you could hear the excitement in this little patient community. But in striking contrast to families' optimism, the program sponsor became very quiet. And after a while, we began asking questions, and the reason became clear. The company didn't plan to move the program forward, at least not for a while. It was paused after considering the complexities and the cost for the next stage of work, even though there was an allowed IND. Optimism was in jeopardy. But hope requires action, and Taylor's Tale revved up its little engine. About a year later, in the second half of 2020, the program was sublicensed. Lost time, lost children, but the program would move on. The rest of our little CLN1 disease community found a way to rev up its engine too, and excitement returned. Within another couple of years, the wheels were in motion for recruitment in Canada. The protocol was released and presented in a poster at the World Symposium, and we believed the sponsor would soon be announcing contacts for enrollment. Neurologists caring for children who were well within the eligibility range were in touch with the PI in Canada. A family in Australia was proactively preparing in hope of a chance to be screened for the program. They were getting passports, making plans for jobs that were going to need to be on hold for an extended period of time. The family made regular calls to BDSRA Australia. Is there any news on the enrollment? Time ticked by, and the mom called again. Is there news? Any news at all? Her child was beginning to lose trunk control, and walking was required to participate in the study. When? Then optimism collapsed. A member of the Batten Disease Association made the call to this young mom to let her know that the trial was paused, with no word about next steps or when it might resume. The depth of this mother's despair was uncontrolled. She literally fell out of view on the screen as this BDSRA representative looked on, utterly helpless in that moment. The mom's reaction was visceral. Hope for her child and this community had been ripped away in the space of just a few words. CLN1 disease was not the only program for a very small patient population that was shuttered in 2021-2022 timeframe. What happened to the CLN1 disease community wasn't unique. Taylor passed away in 2018, yet I was still crushed by the news that progress and hope for this little community was halted once again. Once you've lived with Batten disease, you know that future children and families must have better options. Can we change slides? From a larger Batten perspective, the CLN1 disease, excuse me, the CLN1 disease program was not the only Batten subtype that suffered a loss during this period of downturn. Take a look at this slide, which shows the Batten community's loss over 3 short years. And when I look at this slide, it literally hurts my heart for a community that I consider to be extended family. For a little context, there are 14 subtypes of Batten disease, representing collectively about 14,000 patients in total. The hexagons with the asterisks are international, programs that are international. The hexagons with the strike-throughs are on hold, and then there's one program that there's one program that represents an investigator-initiated study. Today, and there should be a strike-through with an asterisk on CLN3, because CLN3, while it's on hold, is actually an international study. Today, May 21st, 2024, there are only 4 active programs in the United States for 3 subtypes of Batten disease. One of those is an N-of-1 investigator-initiated trial for CLN1 disease, the program that Taylor's Tale initiated in 2013. And that was almost 12 years ago, and it's 1 child. 11 programs trimmed to 4 in 3 years' time. The Batten community has learned from painful experience that as soon as tomorrow, this could change again. Will we learn about another program getting halted, another company strategically reprioritizing its pipeline, another carefully crafted press release to appease and assure company shareholders while crushing the hearts and hopes of an entire community? We've learned from painful experience that the clinical trial landscape continues to be volatile, unpredictable, and yes, ruthless, particularly for very small populations. Even as investment in rare diseases is beginning to wake to a new spring, patient communities, particularly those that are very small, have lost trust that the system can work for them. Hope and optimism may present the deepest loss of all. These small patient communities are damaged and bitter, and surely you can understand why. To know there is science with the potential to improve life significantly, yet forced to watch it shut down right before your eyes. These are real people pulled to the edge of hope only to be pushed from the cliff. The path forward for the Batten community seems murky right now. As an example, there's concern regarding the regulatory requirements across Batten disease trials and subtypes. Whereas natural history data was used as a comparator in the pivotal trial of serlipanase alpha ERT for treatment of CLN2 disease, A one-to-one placebo-controlled study is now required for a forthcoming phase 3 study of miglustat in CLN3. And miglustat's a drug that's already approved for another condition, even where experts argue that sufficient natural history data exist. For a patient community, these inconsistencies can sometimes feel unethical and unfair to the families who've already faced an unimaginable diagnosis, who are losing their child little by little every day with no prospect of hope. To be enrolled in a clinical trial and be given the tantalizing whiff of a potential treatment, to go through the all-consuming process of clinical trial participation, only to find out sometime later that their child had in fact been on a placebo the whole time. For one moment, I want you to think about what this must feel like for a parent. It's another blow on the journey of relentless, recurrent losses in their child's devastating and life-limiting illness. Consider the time and the cost involved in running a trial that's effectively twice the size and magnitude with a placebo-controlled arm. The trial is on hold. The CLN3 trial is on hold right now as the sponsor works to acquire additional funding. The study will have to be larger. The study's going to have to be larger with the placebo-controlled arm. That they're being forced, and that means they're being forced to take on 9 sites instead of 9 sites across 7 countries to get that kind of patient engagement. For small companies and organizations like Beyond Batten Disease Foundation, which sponsors these studies, it becomes harder, longer, and more complex to navigate, perhaps prohibitively. Beyond Batten Disease is led by the father of a CLN3 child, and he created this group to drive progress for CLN3 Batten disease. And it's a very small population. There've got to be better ways to bring therapies to our children and families who are just begging for a chance to live better lives, but faced with the realities of being part of a small population. Quickly, because I'm running out of time, I want to restate the statistics that I know all of you in this room know. 30 million Americans, and that's 10% of our U.S. population, living with about one of 10,000 rare diseases. Approximately 40% of those 10,000 rare diseases have 50 patients or less. Using traditional models, drug development for these rare disease, these ultra-rare populations becomes extremely challenging. If we can't find better solutions for these patients and families, millions of people, like my daughter, will continue to be left behind without hope and optimism that they too can have a better chance at a better life. Before we can fix something, we need to define it. And we need to set measurable parameters. Right now, there's no true distinction between a rare condition that affects a few people and one that affects 199,000 people. Without a consistent definition for what constitutes an ultra-rare condition, it seems illogical to me to talk about pathways to treatments and cures if we haven't fully defined the problem. It's not going to be an easy solution because it comes with the potential for great risk. However, if we do it well, it'll provide great benefit and the chance to improve outcomes for people like Taylor. And I'm willing to take that leap of faith. Are you? So I'm looking forward to the discussions we're going to have today. I don't have all the answers, but I know that we have to address the problem. So let's break down the barriers. Let's create efficiencies to deliver these therapies to those in need, and even those living with the rarest of rare conditions, because they deserve hope and optimism just as patients and families faced with more common conditions. We can bring treatments to them safely, efficaciously, and sooner. hope is indeed informed optimism. And for this story, informed optimism begins with better defining those left behind. Thank you.
Great. Good morning. Hi. Thank you for providing me with an opportunity to share a few words regarding the drug development paradigm for rare disease. And I'm here to represent BridgeBio. My name is Amanda Malakoff, Senior Director of Government Affairs. More importantly, thank you for letting me step in for Adora Endu, who really wanted to be here today. So as most of us here in the room know, the statistics that we so often hear don't lie. Rare diseases present global health crises with dire consequences. In 2021, for the first time, the UN General Assembly adopted the first ever resolution on addressing challenges of persons living with a rare disease and their families, with the hope of making a difference for the over 400 million people suffering from a rare disease globally. The resolution was adopted by all 193 UN member states and is the first UN document to recognize the specific challenges of people living with a rare disease. Here in the US, as many of us also know, 1 in 10 Americans are impacted by a rare disease, which means that many of us here in this room either have some form of a rare disease or know someone impacted by these diseases, whether friends, family, colleagues, or neighbors. There are now over 10,000 identified rare diseases, which is staggering. What's staggering about this statistic is that 95% of these diseases have no available treatment. And with 95% of diseases with no available treatment, many of us are here in this room today to ask the question, why? And collectively, what can we all do to change this? At BridgeBio, we, along with other industry biotechs, believe that we have a meaningful role to play in changing the trajectory for the rare disease community. We are a Bay Area biotech company founded in 2015 with the goal to discover, create, test, and deliver transformative medicines to treat patients who suffer from rare genetic diseases. In 2021, just 6 years after the company was founded, we received our first 2 approvals, which is notable. And what is notable about that is that both approvals were for ultra-rare diseases. The first was for molybdenum cofactor deficiency type A, an ultra-rare metabolic disorder presenting in the first few days of life, causing intractable seizures, brain injury, and death in early childhood around the age of 4. The approval was based on clinical trials that showed a meaningful increase in the overall survival compared to the natural history of the disease. The second product was approved in the same year for another ultra-rare disease, FGFR2 fusion cholangiocarcinoma, where approval was granted under the accelerated approval pathway. While our unique business model and scientific approach of targeting the disease at its source has enabled us to quickly develop these 2 treatments effectively and efficiently for patients, the reality is that both programs required extensive collaboration with FDA and the ability to leverage regulatory flexibility given the rarity of the diseases. As mentioned, the first approval leveraged natural history as a comparator instead of a placebo control, and the second leveraged accelerated approval to bring the treatment to patients. Arguably, neither of these treatments would have been made available if the necessary regulatory flexibility was not applied. And these 2 examples do not happen as often as we need them to. More often than not, programs are ended prematurely due to insurmountable complexities present in the development and regulatory pathway. It is well understood that all rare diseases will require incentives and regulatory flexibilities to ensure treatments are developed. We must remember that in the 1970s, prior to the passage of the Orphan Drug Act, there were only 10 drugs approved to treat rare diseases, 10. While the ODA has spurred innovation in rare diseases more broadly, we do need to think about whether more is needed to ensure that all categories of rare disease have a development and commercial path. There is no clear definition for the categories of rare disease, one of the reasons why we're here today. But for the sake of and ease of discussion, we've placed them into these categories: rare, ultra-rare, and N of 1. Rare representing all diseases impacting less than 200,000 people. Ultra-rare, depending on which reports you read, it could range from diseases impacting less than 2,000 to 10,000 people in the US. What is interesting is that reports suggest that the vast majority of rare diseases are considered ultra-rare, especially with advancements in precision medicine. An analysis done by our friends here at EveryLife found that about 85% of rare diseases occur in fewer than 5,000 US patients. And then N of 1 impacting people in the single digits. Now, we know that rare disease complexity and the application of the existing regulatory framework can present challenges in development and are especially impactful for very rare diseases. Rare disease heterogeneity and limited natural history, lack of regulatory precedent, no clear path for novel surrogate endpoint acceptance, randomized placebo-controlled trials often expected, inconsistent use of the accelerated approval pathway, and heightened focus on feels, functions, and survives clinical endpoints. Now, these challenges are even more apparent for ultra-rare diseases and can be devastating for programs. The result of this is that the vast majority of compounds are not progressing into clinical development, and certainly not to the bedside to the many people that are waiting. At BridgeBio, we have a broad portfolio that spans genetic diseases of different sizes. We continue to advance development in very small populations to provide patients with treatment options for some of the most devastating diseases. For example, we're currently developing a treatment for LGMD2I, a genetic disease caused by mutations in the FKRP gene that is found throughout the body, especially in the brain, heart, and skeletal muscles. This disease causes weakness in muscles close to the body, which can cause difficulty walking. Calf hypertrophy, dilated cardiomyopathy, and respiratory difficulties are also seen. We are also developing a treatment for another ultra-rare disease known as convened disease, a rare neurological disease that affects children from birth and has no treatment options beyond supportive care. As we advance development for both programs, we recognize that innovative clinical trial designs, leveraging single trials, given the very small populations, and the ability to leverage novel endpoints, including biomarkers, to support accelerated approval, incorporating patient experience data to help regulators with the interpretation of small data sets, and approaches to, sorry, I just realized I should go to this slide. And approaches to leveraging natural history data are all critical to ensure development. Essentially, the regulatory tools available to regulators must keep pace with scientific advances in order to enable treatments for such ultra-rare diseases to be developed quickly. With this in mind, it is imperative that Congress enable a regulatory infrastructure to accommodate the needs of such diseases. So I know one of the things we want to talk about today is opportunities for policy improvement for ultra-rare therapy development. So we suggest a few topics that could help support the development of more ultra-rare disease therapies and open this up really for discussion throughout the day. But our first call to action from a policy perspective would be making sure that we all work together to see that the priority review voucher program for rare pediatric diseases is reauthorized. This program, as many of us know, is critical to offset the R&D costs for very small populations that otherwise may not be commercially viable. So we really want to see Congress pass the Creating Hope Reauthorization Act and renew the program. In addition, we propose that a new framework for primary disease activity biomarkers, as suggested by Emil Kakkis at Ultragenics, be advanced to enable a consistent framework for leveraging biomarkers for accelerated approval, and we think this would greatly improve the development of therapies for rare and ultra-rare diseases. Next, we suggest a new idea that we're terming science-focused drug development meetings, really with the goal and the purpose that with over 10,000 rare diseases, it's absolutely critical that there's an ability for FDA to engage with experts that understand the evolution and the fast-moving science behind specific rare diseases and can help educate FDA staff who are making the regulatory decisions. So we suggested a new meeting type to make sure that FDA can systematically obtain the scientific information they need to make these informed decisions on rare diseases. And then lastly, we recommend that Congress move forward with the Benefit Act, which would ensure that patient experience data, PFDD-related data, are considered as part of the FDA's risk-benefit assessment. So these are some ideas and framing for today's discussion. We really are glad to be a part of it and look forward to working with all of you to advance this conversation forward. Thank you.
Hello, I am Janet Maynard. I am the Director of the Office of Rare Diseases, Pediatrics, Urologic, and Reproductive Medicine at the FDA. It's such a pleasure to be here today for this scientific workshop that the EveryLife has organized, and thank you very much for this opportunity. So today I'll be touching on some of the regulatory considerations related to small population drug development. This is just a standard disclaimer and disclosure slide. This is not intended to convey official US FDA policy. So thinking from perspectives, at CDER, the Center for Drug Evaluation and Research, we are really dedicated to ensuring that there are safe and effective drugs that are available to improve the health of patients. And I really see this as such an exciting time for rare disease product development. There's so many scientific advancements. We have just so much momentum in the field. And I see it as really an optimistic time where we have the ability for therapies to potentially really change the lives of patients. I am a rheumatologist by training, and so I've been lucky during my clinical career to see how the availability of many therapeutics for a variety of rheumatologic illnesses has really transformed the field and really changed the trajectory of the lives of many patients. And I say that in a positive way, to say that really I think working together and having collaboration, that's so important to propelling a field forward. And I see that as an important time that we are in right now in rare disease product development. And we all need to keep focusing on our common goal, and that's really to have more safe and effective drugs available for more patients more efficiently. So this slide shows sort of where we're going in terms of the number of orphan approvals that we're seeing over time. And in the blue bars, you see the number of non-orphan novel approvals by CDER, and then in green, the number of orphan novel approvals. And it shows on a yearly basis what's happening in terms of the number of approvals. And you can see over time in the purple line, which is the proportion of orphan novel approvals of all approvals, that there's been an increase in the terms of the proportion of orphan novel approvals each year. So we're definitely making progress. We're seeing that there are more therapies available for patients. But I know for all of you in this room and online that you're like, but there's still so much work that we have to do. While we still have more therapies available for patients, clearly the vast majority of patients with rare diseases still do not have safe and effective therapies available to them. Just touching on some of the challenges that we just heard, I think we have alignment on some of the challenges I think that we're facing in rare disease product development. And I think one that we're going to focus on today is really about population size. So clearly when you have small populations, it can make trial design considerations extremely difficult. Also, when you have a given rare disease that might have significant heterogeneity, possibly in terms of the disease manifestations, that can be challenging as you try and think about, well, what's the best endpoint that we should assess in this trial, and what would be the most meaningful to the majority of the patients that has this illness. Also, unfortunately, for many rare diseases, the natural history may be poorly understood. And this is really foundational when we're thinking about rare disease product development. Because if we don't know the natural history, it's extremely challenging to design a clinical trial, such as the length of time that we should wait to see a potential change in an endpoint. As we know, these diseases are often serious or life-threatening. The majority are progressive, and unfortunately, approximately half affect children. So there's really urgent needs for therapies for rare diseases. Many drug development tools are often lacking for rare disease drug development, and as you just heard, there can also be a lack of regulatory precedent, which can make it challenging as you're designing your clinical trial. So within CEDAR, we recognize these challenges, and we're really working so that we can collaborate with the community as we think about ways to address those challenges. And in 2022, we launched the Accelerating Rare Disease Cures, or ARC program. And this slide just shows some of the initiatives that ARC is involved with. ARC is a CEDAR-wide effort, and we're really trying to leverage all of the different activities that are going on in rare disease product development across CEDAR. And as you can see, there's many different initiatives, and it's helpful to have ARC, which is sort of an umbrella that helps connect all of these different initiatives that are going on in rare disease product development and really synergize them and help facilitate rare disease product development. I think another important aspect of ARC is also scientific and regulatory innovation and advancement, because I think understanding translational science is really foundational to our rare disease product development. And also, as part of that, making sure we have outreach and education, both internally within FDA and also with our external stakeholders, where we can hear directly from the community about the challenges that they're facing. And I wanted to highlight one of our recent initiatives. It's the LEADER and LEADER 3D initiative, because I think it's relevant to some of our conversations today. As part of this initiative, we conducted interviews, and also there was a public docket. And we were really looking for feedback on what are the challenges that you are experiencing in rare disease product development directly from those who are involved. It was incredibly helpful to get that feedback, and I want to thank everyone online and in the room who contributed feedback. We recently released a report, and really the goal of this is to highlight ways that we can continue to advance rare disease product development through additional education and outreach activities at FDA. I also wanted to highlight a new CEDAR initiative called C3TI, which is the CEDAR Center for Innovation, which really is trying to help us harness all that's going on in innovation in CEDAR and bring collaboration together, because as we use more innovative clinical trials, this will help enhance efficiency in product development. This initiative is not specific to rare diseases, but I think it's very relevant as we talk today about innovation and the importance of innovation in terms of making our clinical trials work for the challenges that we are facing in rare disease product development. And then when we think more about sort of the review activities, I also wanted to make sure everyone was aware of the recently announced advisory committee, the Genetic Metabolic Diseases Advisory Committee. So this advisory committee is specifically being set up to advise on topics that would be in CEDAR related to rare genetic metabolic diseases. And as you are all aware, there are specific challenges that we face when we're thinking about drug development in this space. They're challenging considerations related to small populations, translational science, the treatment of inborn errors of metabolism. And this advisory committee will really serve as a way to bring together experts to have those discussions about challenging applications that we're reviewing in this space. So in conclusion, I really am optimistic that we have the potential to really advance treatment options for rare disease patients and families. We know there are challenges, and we're really looking for ways that we can collaborate to address those challenges and appreciate the opportunity to have that collaboration, because I think it's truly key to advancing our shared common goal, which is having more treatments available for more patients more efficiently. Thank you. I'm going to stay up here. Okay.
Thank you very much, Dr. Maynard, and you get your choice of chairs. I'd like to call up Ethan Perlstein, and he'll serve as the moderator for this next panel, and also the other panelists will come forward, Sharon, and then Aaron and others will self-introduce as you will.
Great. Hey, everybody, how's DC doing this morning? All my beautiful people on the live stream, hello, thank you for tuning in. Let's get up on stage, panelists. All right, so I'm Ethan Perlstein. I'm a scientist, biotech entrepreneur, and investor. I'm the founder and CEO of Perlara, the world's first biotech public benefit corporation, and also co-founder and CEO of Maggie's Pearl, which started out as a joint venture 50-50 between Perlara and a incredible rare disease family, like many of you in this room, from Sturgis, Michigan. So why don't we start to my left here, or where should we begin with introductions?
Sure. Tim Franson, failed retiree.
Oh, you have a mic. Everyone should have a mic. So please project.
I think we have to share.
Yeah, hello everyone. Sharon King, Taylor's Tale.
Hello, everybody. I am Paul Melmeyer. I'm the Executive Vice President of Public Policy and Advocacy at the Muscular Dystrophy Association. Grateful to be here.
Good morning. I'm Aaron Goldenberg. I'm a bioethicist and vice chair of the Department of Bioethics at Case Western Reserve University in Cleveland, Ohio, but most of my work centers on newborn screening and rare disease and ethical and social and legal implications.
And I'm Janet Maynard from the FDA.
Hi, everyone. I'm Sal Race from T. Rowe Price. I'm one of the biotech analysts and run one of our healthcare funds there.
All right, let's start with an opening lightning round question, and maybe some people can't answer this specifically, but let's address the numerical elephant in the room. What should be the numerical cutoff for ultra-rare? And if it's not a numerical cutoff, is there a collection of attributes or characteristics that would uniquely define ultra-rare?
Interesting challenge, Ethan. And when we talk about redefining elements, one thing is numerical, the second thing is severity, because I think we're all challenged with how do we prioritize across so many needs and demands, and how do we attempt to rank order so that we have some sense. I don't know what the right number is, and quite frankly, when you look at orphan diseases, the numbers for the EU and World Health Organization and FDA are all different. They're all in the same kind of ballpark, but there's hardly consensus there. So I think we need to work on criteria, which includes severity as well as numerical burden.
Any other criteria you would call out, or those are the two big ones?
I'd start with those.
Great.
And I would agree with that. But I'm really reticent to just arbitrarily pick a number. I would say for those diseases that are really struggling to be commercially viable. But again, we've had approvals for very small diseases, very small. So it goes to show, there is flexibility there. So how do you arbitrarily pick that number? Is 10,000 the right number? Is it US focused or is it global? Again, I think for me, are we looking at progressive, life-threatening illnesses? I also look at the severity of the disease. I think about what my daughter lived with. Over the course of her disease, she lost her vision. She lost her ability to walk, to talk, to swallow. She had suffered seizures. She suffered cognitive loss. Bat disease is a really devastating, monstrous disease. And we need better options for these families.
So for my answer, just for some context of why I'm even up here in the first place, we at the MDA, we serve the rare neuromuscular disease community, and that includes communities of just a handful of individuals, or even just one individual that's been discovered with a specific genetic mutation, all the way to populations such as the Charcot-Marie Tooth population that's actually one of the larger rare diseases that still fit within the rare disease definition. So we kind of have to approach it from all directions, if you will. So my answer, as I answered the poll question just about maybe an hour or so ago, was unsure. And the reason for that is that we currently, at the very least, lean towards trying to pursue policy interventions that target the issues, that target the problems of ultra-rare therapeutic development, regulatory consideration, and commercialization and access. And we know that these problems don't start and stop at a specific number. There are populations that might be 10 or 20,000 individuals that have these challenges. There might be populations of 500 people that don't, for that matter. So we trend towards looking at the issues in policy development and less towards looking at a specific numerical cutoff.
I will follow that similar trend of being unsure. For me, I think I want to introduce another criteria, or at least another consideration, which is one of equity, that I know is something that a lot of you value and think about a lot in your work. We tend to spend most of our time talking about equity and disparities issues between rare and common diseases, but there are lots of equity issues between rare conditions, conditions that have more power, more money, more voice. And I think as we consider what ultra-rare might look like and trying to either pinpoint a number or a criteria for which inclusion in ultra-rare is considered, I think we need to think about how that decision may either help those disparities, those equity questions, or actually exacerbate those disparities questions. And if we can figure out a way collectively to utilize that definitional movement for reducing disparities, for increasing equity between rare conditions, then I'm all for it. I think that's a great thing. But I think we need to be very careful and cognizant of the potential for those kinds of definitional moves to actually worsen some of the disparities, especially for rare conditions for which families and communities have less voice, less empowerment, less resources.
Yeah, so I can't comment specifically on potential legislative changes, but I can think sort of more broadly about the challenges we see in rare disease product development. And this sort of builds on what others have said, that there are so many challenges, and it's so specific to the individual situation, and it's very important that we consider those issues, such as disease severity, whether or not there's an endpoint. Do we understand the natural history? I think those are all so important when we're framing what each rare disease drug development program needs to be successful. And I was thinking back this morning to sort of when I started speaking about rare disease considerations, and at that time I was really emphasizing commonalities. And I do think it's very important that we think about those and the commonalities we have, but also I realize it's more nuanced than that. When we think about commonalities, it's also to understand the specific situation that's before us. What are the specific needs of the rare disease community or program that we're working on, to really understand that, because leveraging that information really, I think, will be beneficial as we think about how to propel the field forward.
Severity is a key factor here, but I also think that having a number, a higher number, wouldn't be such a bad thing. And that allows for greater inclusion. It allows for greater equity. And so, I think if we move towards a path of having a number for rare disease, a higher number that is more broadly collective of the unmet need, moves in the right direction. And so I don't think any of us know what the right number is, but let's say a number like 50,000, would that help move things more quickly and in the right direction? I think there's a chance. Now, obviously we have to have flexibility. So let's say you have a condition that has 60,000. Does that mean all of a sudden that needs to be excluded? No. So I think a range, a higher number, and flexibility to be more inclusive could move us in the right direction.
So some of you took the bait, but I guess I would throw out 2,000 as the cutoff, and I'm just echoing ultra-rare drug developer extraordinaire, Emil Kakis, and he gave a really nice talk and made a case for an ultra-orphan drug act and picked 2,000 as the number. And I think there's some elegance to that, given that 200,000 is the ODA definition, and we're talking about sort of the 1% of this population that actually represents 99% of all rare diseases, because it's this massive long tail. But we have all talked about the challenges of ultra-rare drug development and the barriers, and there's so many aspects from discovery to the clinic. I would like each of you, based on your lived experience, to talk about one of the major barriers you experienced in ultra-rare drug development. Yeah, just to mix it up, why don't we start all the way on the left?
Oh, sure. I'm a biotech investor, and when we're making investments in private biotech companies, the first thing we always ask for are the regulatory documents. And it's critical because really, like, there isn't a path forward if there isn't alignment from the company with what the regulators are asking for. And I think specificity is a big challenge here, because often what we've seen in regulatory documents are, well, you could try this, but it's at your own risk. And I think if we can move towards having greater clarity on what the bar is, what the requirement is, with room for a lot of dialogue to agree on what that bar is, I think that can significantly move things forward in a much faster fashion. Because from our side, what we often see is a little bit of shift in the accountability. And what happens is when the feedback from the regulatory bodies is that, well, you can try this, it's at your own risk. What that does is it shifts all of the risk to the patients, the companies, and the investors. And I think greater sharing of that accountability by saying, well, why don't we agree on what the right markers are, what the right preclinical models are, that would allow for, essentially sharing that risk in what we all agree is a very high-risk endeavor. And so I think pathways to come to agreement on the right bar for each individual indication, and an agreement that if that has been met, there's a clear path to bringing the drug to market. I really, from my perspective, I think that's the biggest challenge, and I think making advancements in that area could hugely benefit all stakeholders.
So there are a lot of potential challenges to mention, but because I was told to pick one, I'm going to pick endpoint selection, which I think is a common challenge in rare disease product development in general, not just to very small populations, but it can be very challenging to pick an endpoint because within a clinical trial, we want to know whether or not a treatment is effective, but we need to have an endpoint that's sensitive to change, that is measuring things that are meaningful to patients, that will also change within the time course of a clinical trial. And so it can be really challenging to pick that endpoint. And so we recognize at FDA that can be a challenge, and so we do have programs, especially the Rare Disease Endpoint Advancement Pilot Program, that are working to facilitate that, but I recognize that is an important challenge within rare disease product development.
So I'm an ethicist. I work both in kind of research ethics and regulatory spaces and in kind of ethics and social spaces. I'm going to separate out my role from a regulatory perspective from an ethical one for a second and not really speak to the regulatory challenges because there are many. I want to talk a little bit about ethics and values. And what I would say is that my work within the rare disease community has shown how frustrated I am and others are that even the ethics literature, the ethics community, hasn't adequately and insufficiently taken into account the values and the voices of rare disease families. And the ethical guidance that we use to formulate research ethics, the way we formulate what is ethical or unethical within the context of clinical trials and other kinds of research, I think also insufficiently take into account the real lived experience of rare disease families and what that means for moving drug development forward. So for me, I think, I'm going to say we as an ethics community, we as a social science community need to do a better job of listening to those voices and integrating the experiences and needs of rare disease families into the way we think about what is ethical and how do you conduct both effective and ethical research within the rare disease community. And we just need to spend more time doing that. We need to do that together, and I think there is a great opportunity. There's lots more of us within the ethics community doing rare disease research, and I think hopefully we can move some of those conversations forward that will help to not only build momentum, but build a framework, a better framework for ethical research within rare disease, and in this case, within ultra-rare disease.
So the one that I will choose is commercialization. Within rare neuromuscular conditions, the vast majority of them are genetic. I'll call out limb-girdle muscular dystrophy type 2C as an example. There are probably about 30 or 40 individuals in the entire United States with this limb-girdle. And it's a monogenetic condition. Gene therapy should be incredibly promising. And yet, for a company to approach a gene therapy for LGMD2C, they may treat the entire population within the clinical trial before it even reaches the market. So that is about as unattractive as it would get, I would imagine, for a biotech or for an investor, looking down at the left side of the panel here, to invest in. And so there's just no commercialization argument, frankly, for a disease of that nature. So that's the one that I would call out. Commercialization is an issue to address within ultra-rare.
You just stole my call out.
There are many more, Sharon. Please pick another one.
Yeah. Yeah, I really want about 10. So, but I would say, so I've been participating for the past year and a half, 2 years, with the Bespoke Gene Therapy Consortium, and I'm participating on the Patient Engagement Committee. And one of the things that I've been, I wanted to say, and I was going to say it in my comments, and then went over my time. I think when we listen to patients, one of the things that I've learned through participation on that committee, I knew it, but it's how we all have to work together to serve these needs of these patient communities. I don't think we're going to make this happen if we can't step out of our boxes and work collectively. But one of the things that I've seen happen within the Bespoke Gene Therapy Consortium is this focus on patient engagement. Because if we really listen to the needs of these patients and families, we're going to know the path we need to take. It may not be clear in the very beginning, but I say we listen to the families, the people who are living with these conditions, who know things that are happening within their homes as they watch their children and their loved ones live with these often severe, devastating illnesses. I think we'll know where we need to go, and we'll be able to answer the most difficult of questions. But it's going to require bringing them to the table and letting them talk about things that we only imagine that's happening in their homes every day, living with these conditions.
Well, I wanted to go last, so I didn't steal the easy ones. And I think I would put top of the list control groups. I think that's probably one of the most frustrating things to patients and their families. It's one of the most confounding of nature to sponsors, manufacturers. And what we can do right now in terms of using artificial intelligence and certain collaborative approaches with Bayesian statistics. And those things are actually going on now between FDA and the Critical Path Institute to look at simulation of control arms, understanding that with disease heterogeneity, there's so many complexities. How do you begin to adjust for that? And when we can learn to refine that more, we reduce the number of actual patients who have to be in control groups. And that facilitates then more patients in the test subject area and is also less of a disincentive to families. So I think we have some real opportunities there.
So how much regulatory flexibility exists on the books today? So we're all pleading for an ultra-orphan drug act and other considerations, but folks out there are not waiting, and so they're hacking the system. For example, taking advantage of single patient INDs, and so I've seen cases of a first single patient IND, and then a second, and then a third, and then a fourth, and then by the fifth or sixth time, the FDA's like, what's going on here? So can you talk about what flexibility already exists today that we can take advantage of, or that has been taken advantage of, either you know about or you've heard about? And let's just kind of go sine wave.
Well, it's a great question, Ethan, and I think flexibility is something that's typically not on the books, but it's something that's interpretational. And being able to say, where are areas that we can look and provide some decisional latitude without changing the standards for approval for substantial evidence. So I think being able to take those kind of interpretive approaches is best data mined from the approvals that have already been successful in taking things just a little bit different than what a straight line review might be. And I think there are precedents. I know Frank Sasanowski's here and has actually written with his colleagues 2 Sentinel papers, including all the examples for that. I think that it's probably required reading for the rare disease community to look at as roadmaps and precedents.
I believe we have approved therapies for very small conditions. How do we make that happen for more? Is it that we don't have the natural history data? How can we make that happen? So we know the flexibility is there, but we're watching, as I mentioned in my comments, we're watching members of the Batten community, subtypes of the Batten community struggling to move forward, to lose so many. And why is that? Is it that we just can't? It takes so long. There's so many complexities to moving forward for these small populations. I'm looking at the CLN5 community. They have a clinical trial in process, and and it's an international trial, and yet they are struggling to find patients to keep the clinical trial going. They're still enrolling. So, how do we look at moving these trials forward for these very small conditions? Are we going to be eventually everything's an N of 1 for these very small?
There are a number of flexibilities to give FDA a shout out for, frankly, in which they've been moving in that direction. Just to name one so I don't steal any from the left side of the panel here. Use of accelerated approval, frankly, especially within the Center for Biologics, what we're seeing there. The direction of using accelerated approval for gene therapies, we saw this with Duchenne last summer. We're hoping to see this a lot more within additional populations in our community. So they're moving in that direction, and that's a flexibility that deserves a lot of credit.
I'll just say that in my world, there are a number of studies going on right now on the ethics and regulatory concerns related to N of 1 research. I think there isn't sufficient work being done at the intersection between rare disease research and N of 1. There can be more, but I do think that the idea of patient engagement, of family engagement, is one that is necessary in order to better assess potential regulatory changes, and something that I know FDA and other organizations are really taking very seriously, which is to listen to families and to do that engagement necessary to make those regulatory functional changes. And so for me, it's encouraging more research like that. It's more of the empirical research within my world, within other worlds that are focused on creating more informed regulatory structures, more informed regulatory choices.
Thank you. I appreciated the shout out to FDA. So I do think at FDA we recognize that what would be feasible for a common disease is clearly not feasible for rare disease product development, and that as a rare disease becomes increasingly more rare, that the complexities can increase. So we do think applying flexibility within these situations is very important, and we've heard some examples of that. Another example would be that most rare disease drug approvals are based on one adequate and well-controlled trial with confirmatory evidence rather than the two adequate and well-controlled trials that usually occur for a more common disease. So we definitely at FDA recognize the need for flexibility, and we are applying flexibility. I do think it's important, though, to remember that we have to apply flexibility to the specific case that is in front of us. So the flexibility needs sort of depend on the considerations of that specific program, and I think that is part of sort of the regulatory science where we really think about exactly what is needed in a specific rare disease situation.
I would echo that. I think we're seeing greater flexibility on the regulatory side. The Duchenne example, I think, is a really important one, and it kind of captures a lot of the issues we're talking about here today. There, you had a phase 3 trial with a very imperfect primary endpoint. Now, the totality of data in that trial showed pretty significant clinical benefit, and we saw a accelerated approval, but with a kind of restricted age range. Now, we have a PDUFA date coming up in June for the possibility of a broader approval in that setting, and I would echo that certainly from the biotech community, the focus there on that outcome, I'd say it's probably the most critical event that's being watched this year, because I think it's a really important data point on kind of where are we in the, the evolution of regulatory flexibility. Because again, you had a phase 3 trial that technically missed its primary endpoint, but showed in totality good evidence. And so I think how the regulators, not just FDA, the EU regulators and international regulators, how they view that totality of evidence and whether they're willing to provide broader access to a medicine like that is going to be a really important data point. But overall, even the fact that we saw the accelerated approval last year, I think is a very encouraging sign.
All right, let's talk about checks, as in investment checks. Sharon, you said that you were sensing that the spring is back in terms of funding and opportunities. Could each of you comment? Let's maybe start with Sal, apropos your background. What is the fundraising climate now for ultra-rare? What does it take now to de-risk an asset? Can you just talk about the general conditions and funding climate?
I think overall the environment has certainly improved. Like if I think back to 2 to 3 years ago, the kind of capital raising environment and the willingness, the risk appetite to invest in, ultra-rare is probably the highest risk area from an investment perspective. And so from that standpoint, I think the willingness and desire to invest in this space is as high as it has been in the last few years. Now, we still have a lot of room for improvement. And, again, I think coming back to, I think we're all in agreement this shared approach is really going to be critical. It's going to be critical for patients, companies, regulators, investors to all be able to collaborate and come to an agreement on, what's the right path forward in each individual indication that both can be sufficiently fast, but also captures the benefit risk that we all want to ensure gets captured. Because the worst thing that could happen is a medicine gets approved and then we learn about, very substantial safety issues. And so it's a very tricky balance and varies setting to setting, and certainly cognizant of how difficult of a job that can be from the regulatory side. But I think the willingness and interest to move forward and raise capital to do this as quickly and robustly as possible is, as good as I've seen in many years.
So I can't speak to checks, but I can speak on sort of rare disease product development from the perspective of what I'm seeing at FDA. And there's definitely so much increased interest in rare disease drug development. And I think what's exciting to me is that that is really occurring across therapeutic areas. And I think that's important because there are definitely important learnings from the different areas. And for us to be able to share that both external to FDA and also within FDA, sometimes a drug development program and one indication will be relevant to a completely different indication for reasons that you would not have guessed. But as we're having that increased interest in rare disease drug development across therapeutic areas, I'm seeing that and really seeing that leveraging of information that can be shared.
I'll just say very quickly, this is definitely not my area of specialty, but as someone who works very closely with our university's kind of foundation relations, philanthropic organizations, I have seen a drastic increase, which I think is a good thing, of local and regional funding opportunities for rare disease, and that may not be sufficient for the very large kinds of trials that might be necessary, but I do think it's been very interesting and I think optimistic, and on my view, to kind of see just the upsurge, even in the last 5 years, of regional opportunities for funding for rare disease research, whether those are from grateful patients or families or other family foundations, but it just seems to be growing, and I think that that's a fantastic thing.
I don't think I actually have anything particularly incisive to say about this, so I'll give Sharon my time.
As I shared earlier, by 2007, Taylor's Tale was investing in research, and we invested in a number of programs. We invested in ERT. We even funded a program at the NIH, and we landed on gene therapy. It's taken us a long time, and while we didn't fund past that initial proof of concept, we've seen this move forward. But it's taken a long time. And I think for investment, when it takes that long a time, I mean, we started funding in 2013. It's really hard for investors to see how this can move forward and to get the return they're looking for when it takes so long to move something forward. And we had that IND, as I shared earlier, 3 years proof of concept, moved within 2 years, there was an IND, and we're still waiting. So how do we expand interest and investment in these very small diseases where there's not going to be a lot of commercial return when it takes so long to get to that endpoint?
Thanks, Sharon. When I think about checks, I think about checks and balances. So one of the things that I think we should discuss is the things that tip investors to positive or negative. And the preservation of incentives is pretty critical. Accelerated approval has been under attack. That's certainly been a vehicle for many rare disease studies for oncology, neurology, and multiple therapeutic areas. Preserving that kind of support, because looking at failed examples, which are exceptions and not the rule, are clearly not the way to educate ourselves. And then I think the preservation of things such as the priority review voucher, which was already reviewed for rare pediatric disorders, is another way to reassure the investment community that they will be able to have a reasonable return and while still providing the value to patients.
So speaking of investment, what does ultra-rare venture philanthropy look like, right? Venture philanthropy was super hot. Cystic Fibrosis Foundation makes all this money, and then everyone's trying to copy them. But they're a whale, and you guys are all minnows. So how does ultra-rare venture philanthropy even work? And if you don't have a comment, you can skip, but I just wanted to springboard on this conversation about investment.
Yeah, I don't know if I should take the bait on this one.
Take it.
But I think that's a distressing question, that those characteristics are hard to identify, so I'm not sure I have a good answer for it.
Sharon?
I'm continuing to think that we've got to find a way to look for these conditions that have, distinct similarities. Can we do them together? Is that platform? How can we bring these conditions forward more quickly that are so small and so hard? I mean, there's not going to be a lot of commercial return. How can we find similarities and potentially prove them together? Can we do them together?
Well, we at MDA are trying. We have invested in research in the past that has led to approved products. Most recently, vamorolone was approved for Duchenne, a product that we were one of the organizations that provided seed funding that now, of course, has resulted in an approval. We're doing similar things within gene therapy. We have a program we call Kickstart, in which we're trying to basically fund all the studies that get up to the pre-IND level to make de-risk that process to then make the product attractive to commercial partners. But we run into all the same problems that we're going over here as well. So, Ethan, we're trying, but it's a challenge.
I think we have to find a way for, good investments to be made that don't require the level of scale that you were referencing. That it shouldn't be just, humongous investment firms or the largest philanthropy VCs are the ones that are able to be successful. Like we have to find ways for, smaller investments to be made that can both be successful, but also ensure that we make good progress on the R&D front. Same. I'd say we're trying. We're working on at T. Rowe Price, we're working on a new healthcare innovation fund that would kind of really focus its efforts on kind of the most cutting edge science in the areas that maybe not everyone wants to be invested in, smaller investments, et cetera. So I'd say, I think we have to find a path where, more individuals, more organizations can actually participate versus leaving it to only a handful of groups to do this process, because I don't think that that's going to be possible over the long term via that approach.
So how do we responsibly move beyond the randomized, double-blind, placebo-controlled RCT paradigm for ultra-rare without sacrificing, standards of evidence, while still being bioethically sort of in compliance with all folks who have a bioethical stake in this? How do we move beyond these placebo-controlled trials for ultra-rare and still convince people that we have interventions that work? We could maybe start from that side.
From my perspective, I think it's two things. We need better preclinical models, and we need better biomarkers. I think we're moving in that direction, but the extent to which we can have preclinical models that are much better at predicting both the safety and potential for efficacy over time, and then also biomarkers that are going to be great predictors of longer-term clinical benefit. But on that front, I think we also have to be in agreement that it's never going to be perfect. Like every good preclinical model is still going to have, it's not going to be 100% predictive, and every good biomarker is never going to be 100% predictive. And that can't be the threshold that we use to make improvements in that area. I think if we can get comfort that, maybe 80 or 90% is sufficiently good for most people, that would make a lot of progress, because if we just have an unreasonably high bar, that's going to lead to really not enough progress, and not enough progress in the speed that which we need to be executing here.
Yeah, I agree that I think translational science is really foundational to rare disease product development. So that's really understanding the pathophysiology of the disease, understanding the target of a drug in a specific disease, and sort of really having that to build your clinical development program, that to me is really important in these rare disease programs. And it does take a significant amount of work up front, and to your point that there can be challenges with non-clinical models. So we need to really think critically about the type of translational science information that we need in a specific development program. And at FDA, we recognize that this is an important aspect of facilitating rare disease drug development. So we have launched a translational science team within CEDAR to help with these considerations. And to your point, it sort of supports us as we're thinking about challenging considerations related to like surrogates or biomarkers. And I think that will be important as we think about tools to support product development.
Yeah, I think this is a really important question. I think part of this goes into what we were talking about a little bit before, which is thinking about different kinds of models, AI models, and kind of other kinds of predictive models that might help us to move away from strictly needing placebo controls. I think it means needing to refocus what ethics of these kinds of rare disease trials looks like. I think we need to be really focused on other kinds of considerations that are important in creating safe, effective, and ethical research. That includes issues around privacy and assuring privacy for families, many of whom may be willing to take on more risks of disclosure of personal information because they want the research to move forward. They want to share what their family's experience was like. But that doesn't mean that we as ethicists or we as researchers should take that kind of privacy any less seriously. Same thing with just kind of taking on risks more generally, where families might be willing to take on higher levels of risk in rare disease research than in other situations. And what does that mean for the ways in which we design our trials? What does that mean for the ways in which we keep ethics central to the informed consent process, to the information disclosure process, to the education process? All of those things need to be built up in ways that have not been as central within rare disease research. And I think there are ways that we can do that collectively, listening to families of what their needs are as they enroll in trials, but at the same time kind of thinking about where families are in terms of their own kind of taking on particular risks. And then we need to, as a community, think about where that tolerance lies in terms of allowing families to take on particular kinds of risks. Those are the kinds of things that I think are more important than thinking just specifically about a placebo or not. I think it's really about trustworthiness within the context of those developing the trials, and thinking about partnerships to develop consent processes and educational processes that are adequately inform families so that that hope is informed, is truly informed, that informed optimism is informed, and that we think about that informed consent process as not just a one-way street, but that informed consent processes are meant to be two-way. They're meant to be interactional. They're meant to be a way for researchers to listen to families. And I think if we build those kinds of structures in and put those as kind of the ethical benchmarks for this kind of research, it allows us to build these structures in ways that maybe don't require some of the things that we have been frustrated with like placebo-controlled trials and those kind of things. It's really about refocusing those ethical benchmarks.
Yeah, just to further that a little bit further from what Dr. Goldenberg said there, the bioethical implication of taking societal generalized values and layering them over very specific small communities that have incredibly different experiences than what the general public, what an able-bodied person, what an otherwise healthy person may be experiencing, and then to be creating policies, programs, approaches based upon the general experience and not upon the individual experience. I think there's a lot of ethical harm that can happen in that situation that we're seeing play out. The other bioethical mention I want to make is that we have the bioethical imperative to tackle the man-made problems that are preventing safe and effective treatments from coming to individuals that otherwise would receive them, that science isn't the issue any longer. That's true for many genetic conditions in which there's a gene therapy that could be developed and could reach our community pretty expeditiously, but due to man-made economic and policy challenges, they're not. And so we have the imperative to be solving the challenges that we ourselves have created. This isn't a scientific issue in some cases. It's a man-made issue. So certainly there's a bioethical issue or imperative there to be tackling those first and foremost.
Thank you for saying that. You said it so well. When I first started back in 2006, I thought that the science was going to be the most difficult part of all. I didn't think about anything else. It was just the science. And then there was this eye-opening moment when I figured out that, the science was moving forward. The science will move forward. And we haven't, in parallel, figured out how we're going to get it to the patients that need it at a time when it can best serve them. How do we diagnose at the right time? And we also didn't start figuring it out in parallel how we were going to get these advanced therapies to the people who need them, how they were going to be able to access it, how we were going to be able to pay for it as a system, as a community, as a country, how we were going to make this work. So it almost seems like science has been the easy part. I said this one time, and I knew that the researcher who had done the early work for CLN1 was sitting in the audience, and I thought, he's probably thinking, you weren't in the lab, you weren't there with me, and you're oversimplifying this. But the truth is, it almost feels like all of the rest of this is the more difficult part. And we have to change that.
I'll be brief, but I think the thing we really wrestle with is when we have unclear efficacy in many of the trials. Safety has not been an impediment when efficacy has been shown, and I think we therefore struggle when we can't really define the effect, when biomarkers are uncertain and so forth. So if we choose to take more regulatory flexibility at that point, we also need a way to pull things back if a confirmatory study shows that the effect, in fact, is not what was hoped. So I think we have those tools now, and how do we better employ them, especially in the rare disease space?
On that note, let's wrap. Everyone, round of applause for this distinguished panel.
So let me just ask all of you to come back in 15 minutes, take care of your virtual work, biologic imperatives, and whatever else is important to you. And we'll see you back here, get a nice drink, and thanks for the great interactions. Sure. Already? There's Esther and her. So folks, if you'll take your seats. We had an interesting morning so far. We heard perspectives from industry, from regulators, from patients, parents. And now what we're going to do is to talk about some of the more process-oriented but critical aspects of clinical and regulatory care. So I would ask Esther Krofa, who's of Milken Institute, to come forward. Well, along with your panel, please. Yep. And they're going to talk about logistical areas as we did in the prior session. We'll have folks introduce themselves and then move forward. So thank you. The floor is yours.
All right. Thank you so much, Tim, and good morning, everyone. My name is Esther Krofa, and I serve as Executive Vice President for Health at the Milken Institute, which also includes Faster Cures, where we've been running a program for over 15 years with 160 patient and disease organizations that are focused on accelerating the development of treatments and cures, certainly as our name implies. So we're delighted to be here for a very important conversation. What I'm going to do is to have everyone introduce themselves. They're going to give 10-minute presentations, and then we'll get into Q&A. We're also going to invite all of you to also be part of our Q&A, so think about some questions that you may have, and certainly as time permits, we'll get to you as well. So let's start first with you.
Thank you, Esther. I'm Emily Milligan. I am the Executive Director for the Barr Syndrome Foundation. Thank you for having me.
Hey, Renee McCarthy. I'm the CEO of Stealth Biotherapeutics. I'm A.J. Allen. I'm the Chief Medical Officer for Institute for Advanced Clinical Trials for Children, or IAC for children.
Justin Hopkin. I was the board chair for the National Niemann-Pick Disease Foundation and sit on their advisory board.
All right, Rainey, Emily, let's have you first start with your 10-minute presentation.
Well, thank you, Esther. If I can have the first slide, please. We want to keep this more conversational, but we wanted to also provide some background. So Barr syndrome is, no matter how we land this plane today or in the future, we are ultra rare. We know of less than 300 people in the entire world, less than 150 here in the US. So we definitely will check that box once we finally determine how to define that. Not only are we ultra rare, but we're also life-threatening. So you can see here from the slide, many of our children in particular, as well as our adults, do die prematurely. And I put this up here to reference, not because, with all of our syndromes, we're all complex. We all have a lot of different clinical symptoms. But I wanted to demonstrate, what are the cardinal characteristics of Barr syndrome, because we can't just have one drug that's going to hit one target. Ideally, we're going to want many drugs that will hit many targets, multiple shots on goals, not just multiple shots on one goal. And that's an important perspective when we start talking about ultra rare and how we define this around feasibility, what's reasonable, because we need to think forward as how we're going to have multiple drugs that will help all of our patients that we represent. That said, we can't let perfect get in the way of good, and we have real problems right now. So I do think we need to be forward-looking in how we progress this conversation while not forgetting the opportunities that we have here today. I also included a couple of words here on the screen that come from our patient-entered registry, because these are ways that we can maybe creatively think of how do we capture what is meaningful in the lives of patients. So for our patients, for example, unplanned hospitalizations, whether they've had a transplant, are they able to eat normally, or do they need to have a G-tube? What are the other things that are impairing them from living the best lives possible for them? So again, Barr syndrome here on the page, but you can replace this for many of the other conditions in this room to talk about common challenges we have to attenuate these multiple symptoms. Next slide, please. So in thinking through, we definitely want a pipeline of opportunities, but we are one of those unfortunate indications that does not yet have an FDA-approved drug. We are close, fingers crossed. And this slide shows our TAS Power trial, and here with Rainey McCarthy, who is the CEO of the company, the sponsor. But from our perspective on the patient advocacy group, I wanted to show you the challenges we have. So 106 people back in 2017, when we started recruiting for a trial, that we knew in this country that had Barr syndrome. You look at the age restrictions, 66 individuals that were over 12 years of age or older. Then 50 people who had not yet had a heart transplant. And then you go down all the eligibility criteria, and we finally got to 16 who we screened, 12 who consented. So if you take the 12 out of that 55, we're talking over 20% of our known population in one trial. And I also wanted to remind people of the statistic here. If you don't yet know this, our prevalence in the US is 1 in a million males. So 106, roughly 300 something million people, we're in the ballpark of who we know is in the country who is diagnosed with Barr syndrome. And going to the data is not the problem. I think this is the title of our talk. And the data is not the problem, is that we have consistently been told, go find more people. They definitionally do not exist. And so as we think about ultra-rare drug development, it's feasibility, it's reasonableness, it's fair, it needs to be equitable, it needs to be appropriate. And these are words I would like us to all contemplate when trying to evolve our thinking, our collective thinking about what is reasonable moving forward. So I'll turn over to Rini to talk a little bit more about this trial and some of the nuances of it.
Hey, thanks, Emily. So ultra-rare, we all know, has a lot of challenges, but one of them is that often you're the first, right? You're the first company, you're the first product to go into clinical development. And often the animal models aren't terribly predictive, and you can take clues from the animal models you have, and you can take clues from other development programs. We were in phase 3 in a related disease of primary mitochondrial myopathy when we started the BARTH program. But phase 2 trials are intended to some extent for learning. And when you're an ultra-rare, you might actually not get a second chance to learn. So in our situation, we didn't treat for long enough in a double-blind, placebo-controlled crossover trial. It was 3 months long. We started to see signals that were, reported by patients, by the doctor, by Barr Syndrome Foundation as being very meaningful in open-label extension, and you're left with how do we move forward from here in an ultra-rare setting. So Emily mentioned, over 20% of the eligible US patient population enrolled. One of the things we thought to do was to turn to a natural history control. And with that, we're well above 50% of the eligible US patient population. There was a very contemporaneous natural history control cohort being collated by Johns Hopkins at the time. It enrolled 79 US patients. When you screen out for over 12, no heart transplants, and longitudinal data, right, because you need 2 data points to compare, we get to 19. But then again, with the interventional patients, we're well over 50% of the eligible US patient population with that. But natural history control studies, also have their limitations. You're not randomizing, right? What is the comparability of the patients? Is there bias, and can you overcome it? There's guidance from FDA to address some of these questions, but again, at the end of the day, looking at what it takes to overcome bias in a patient population of 10,000 or 5,000 versus what it might take to overcome bias in a patient population of sub 130, maybe that needs to be a different scale. And so these are some of the thorny challenges that we've been addressing with the agency. Our natural history control study was positive. The long-term open label data was positive. We did see changes on, endpoints that aren't subject to bias, so physiological endpoints like heart function and the biomarker for the disease. But at the end of the day, what does it take to get to convincing evidence of effectiveness? And is there a path to that in an ultra-rare disease setting? Some of the things that we struggled with in our conversations is it even ethical to do another placebo-controlled trial? I mean, this is a question FDA put to us, so it's not just a sponsor saying this. These were real conversations. Is it even ethical to do if you can't power it properly to see a treatment effect? So I think that when we talk about AI simulations of potential control arms, the potential for external controls, which are recognized as a form of control, and how much weight we can put on that evidence, there are situations in the ultra-rare world where it is more necessary to rely on those types of flexibilities than it would be in a more common rare disease, as it were, where you could probably mount a phase 3 trial or another phase 2, and you could more definitively answer some of those questions. So with that.
Thank you so much. I'm actually going to go to Justin, and we'll come back to AJ. All right, Justin.
Great, thank you.
Yep.
So I'm going to talk a little bit about Niemann-Pick type C. It's a progressive neurodegenerative disorder like some of the disorders you've already heard about today, and I think when you're thinking about targeting the CNS, things become much more difficult. Of course, it's heterogeneous. Age of onset can vary, and the progressive symptoms can progress at different rates. And so designing clinical trials with a heterogeneous population with age of onset that can be variable and progression that can be variable is challenging, but it's even more challenging when you have 200 patients in the entire United States that you're trying to build multiple clinical trials around, and that's been the story for NPC for the last decade. We talked about shots on goal. The first shot on goal for NPC was prior to my engagement with the Niemann-Pick Disease Foundation in 2010. Miglustat had been approved in the EU. It had been approved in Russia, South Korea, and Canada for Niemann-Pick disease type C. It had already been approved for Gaucher. There was an advisory committee hearing which seemed to be pretty positive, but 2 months later, the FDA elected not to approve Miglustat. And the data was followed in a registry that was sponsored by the industry sponsor. Later on, we have seen that there does appear to be a survival benefit as well as a benefit to preventing significant changes such as pneumonias, aspiration, and things like that. So it remains the standard of care, but inaccessible to patients in the United States. One unique aspect, we talked about privilege a little bit earlier to the Niemann-Pick type C community, is the privilege that they have and that they, I think, have earned through a lot of hard work and advocacy at the ground roots level. Their Parsegia Medical Research Fund has raised over $50 million that's been put forward towards research over the last 30 years. There's a national and international patient organization, which are both 30 years old. There's an NIH natural history study, which is available. And there's also an international patient registry. So they've really worked hard as a community to try and put all the everything in place to be a good market for drug development and to have the tools and resources available. Fast forward to 2018, the work of a lot of that research culminated in several different programs developing, and the first study that released its pivotal, or the first company that released its pivotal trial results showed essentially a negative study where the difference between placebo and the intervention arm did not reach statistical significance. I think from a community perspective, this was really surprising because of the known natural history of the disease from what we understood, and seeing patients who had been on this therapy for years and the benefits that at least some of the patients were perceiving. This was a relatively large clinical trial by NPC perspective with 50 patients, but it doesn't take too many patients to throw your P of 0.05 out the door. And there was a need to get in front of the FDA and to deliver this message of what the disease was like and how this treatment appeared to be meaningful along with a lot of the other treatments. So we had a PFDD date scheduled in 2018 to talk about this. And the message was really delivered, I think, well by the patient population that a acceptance of uncertain benefit was completely fine because the known outcome was death. These patients all die by 13 or 14 years old when they are diagnosed in childhood, and so anything is better than what the reality of the disease with. The acceptance of side effects associated with any therapy, but also uncertain benefit, were clearly on display, and I think they did a nice job, and we appreciated FDA's attendance. They were interactive, they asked questions, and I think that was great. 2019, FDA invited us to a listening session to talk about both diseases, Niemann-Pick type C and the other disease. In that discussion, the community discussed alternative trial designs and really thinking about the role of placebo. And I think we've talked a little bit about placebo, and we've also talked about control. And I think it may be important to differentiate the two a little bit. I think if you have an approval and you have a group that's a control group that's getting something that they believe might be impactful, that's acceptable in many community members' eyes. Whereas if you're the first therapy that's going to be approved, placebo in a neurodegenerative disorder where time is neurons, as was said earlier, and time is life, looking at longer controls with placebo-controlled trials is not acceptable. The other thing we also talked about is that there will need to be multiple therapies for a complex disease like this to be approved. There is not a cure that we foresee in any near future. There appear to be therapies that benefit certain patients and certain symptoms, and we wanted to make sure that that message was delivered clearly, that we were going to need multiple therapies to treat this very complex disease. We unfortunately had another company which received a response that was not positive from the FDA. This time, a lot of the feedback that we received and the feedback that's on even the website of the company at this time suggested that it's actually the tool that we used and the endpoint that we used and not necessarily the drug or the outcome. So the Niemann-Pick Clinical Severity Scale, or the NPC scale that we use as part of our natural history study that really is the core to all the research that's been done at the NIH, started with 17 variables to try and encompass the full breadth and variability of the disease. And actually, based on feedback from the FDA and EMA, that tool being used in clinical trials with 17 different variables was probably a little cumbersome to be implemented at a bunch of different clinical trial sites. So the community came together and said, okay, as clinicians, researchers, and patients, what are the 5 most important things? Let's narrow this down. And so we landed on the Niemann-Pick clinical severity scale with 5 measures: cognition, gait, fine motor, speech, and swallow. And each of those is on a 5-point scale. And to get from one point to the other on just one of those, we determined was very clinically meaningful to clinicians and to patients both. And we wanted to use a scale that could identify change in one symptom, because we realized with such a complex disease, if we were going to use one tool across the disease, we had to be able to detect change in different things. And so we were hopeful that this could be the scale that was used. And the feedback when we got that was challenging to think that it was actually the scale that was the problem and potentially not the drug. So then we had another listening session with the FDA on just specifically endpoints in the clinical severity scale. It was a great discussion, good dialogue. We agreed we needed to move forward because we didn't want to throw away decades of data and a tool we're using in active clinical trials. We moved forward with a Duke-Margolis listening session and a discussion session where we talked about the clinical severity scale, but we also talked about biomarkers, and we've talked about digital health technologies. And I think moving forward, we are currently engaged with CPATH to hopefully identify as a group, potentially not just one disease, but multiple diseases. Is there a commonality amongst other diseases that are like ours that we can come together and identify a clinical endpoint? We continue to work very hard with biomarkers and trying to identify a disease-specific biomarker to try, and again, looking forward, have endpoints that can be meaningful and that the regulatory agencies can recognize as sound, but that are also acceptable. I think just talking about where we are today, we're very fortunate that when we started these conversations 6, 7 years ago, we had 4 clinical programs that were in development. Today, we have all 4 of those clinical programs still functional and moving forward. And we're very optimistic that each and every one of those can be impactful. I think if I could reflect back a little bit on our most recent discussions with the FDA and where we're at now and some of the discussions we've had, our response to trying to increase sensitivity has been longer clinical trials with enrolling more patients. And I think with only having 200 patients here, longitudinally, that's not a sustainable way to move forward. And so hopefully with some of the works that we have going on with CPATH, with the FDA, and through Duke-Margolis, that we can, moving forward, have clinical trials with different endpoints, but at the same time, look at the 4 clinical programs that we have now that appear to be very beneficial to patients, and we can get approvals to patients. Thanks.
Thank you so much, Justin. I'm going to come to you now, AJ. We just heard from 2 case studies, right, with Barth disease and Niemann-Pick's, and they talked about the challenges with drug development. That's our topic for this particular panel, which is how do we think about what are the opportunities, but yet how do we overcome these challenges that we just heard about for really small patient populations for drug development? And you've had such a great breadth of experience from this perspective, and certainly with the pediatric population as well. We'll love to hear you reflect and react to what you just heard from these 2.
So it's interesting. Just a brief reflection on something. A long time ago when I started at Lilly, I heard a senior executive, they presented a slide and said, this isn't rocket science, it's harder. And I think in a lot of ways, this is what's true about the rare disease space and pediatrics also, that you're always, it's a lot more difficult than the typical, if you're looking at a drug to prevent heart disease or cure lung cancer or whatever. And it starts with the fact that you just have so many diseases where, as was pointed out by both programs, where you don't have that many patients. So we're almost, one way that I think about ultra-rare diseases is I'm in a situation where the typical randomized placebo-controlled or randomized controlled by standard of care just isn't possible. I mean, you can't do it. And often there's also a secondary question that comes into play, which is, okay, what do you measure? Which was mentioned also, and can be critically important because the instrument that you use to measure something, and usually we're talking about safety or an outcome, that decides the size of a clinical trial that we're talking about. And so in some ways, it's a circular argument. Do we have enough patients to run this sort of trial? Is the outcome something that we can do that, we can get down to enough patients, et cetera? And it just, there's an inherent complexity there, which throw in the additional aspect that because these are rare diseases, many of them may have just been discovered in the last 10 or 20 years, the science is incomplete. And this is often a problem in pediatrics generally, but I think it's especially an issue in terms of rare diseases because, there may not be any sort of, we may not know what the exact cause is. Now, more and more we're having some sense of, is there a particular genetic marker of that, but that doesn't mean we have an animal model in which we can test these things. So there's a lot of sort of feeling and thinking and suspicions and hunches and that goes into this whole process that I think we have to acknowledge makes it a lot more difficult. And the other thing that's true is that while you often have academics and others who may have been involved in studying the disease, coming up with the solution in that have gotten involved in creating a company and trying to develop it, they have a lot of knowledge about the disease, the science and such, but they don't necessarily have a lot of knowledge about the regulatory framework, whether it be just the basic rules around safety and efficacy of drug studies, the pediatric rules, if this is a disease with a pediatric indication, what sort of concerns come into play with the safety in particular can be a big factor because, all of us believe that the drug we're working on is going to work. There's actually some literature on that. And we're all prone to confirmation bias. So there's this inherent belief this is going to work, and yet we don't know whether it will. I mean, that's the whole reason for doing the studies. So we have to, I think, be a little bit careful about assuming those sorts of things. And I think the other thing we have to remember is there's a whole bunch of what I'm going to call economic exchanges or economic markets that come into play. And the value proposition is not the same for everyone. As we heard with the discussion from patients, when you look at the impact on the family, I'm thinking of Sharon's comments here, you look at the impact on the family, and the families are all desperate. They want a cure yesterday because their child is sick or their loved one is sick today, and maybe has a terminal illness. And is this going to be available in time? And even when we look at some of these conditions, things like some of the genetic therapies, the clock is ticking. You're getting progressive degradation, for example, with Duchenne's muscular dystrophy in the muscles. And as that's happening, the window may be closing for a genetic therapy to have an effectiveness. And so you really want to start doing those studies earlier. And that may be, something where you have other forces that are coming into play that are raising questions. Things like the IRBs may be saying, well, how can we put a child that's, say, 4 years old on a genetic therapy that we don't even know works yet, given this circumstance? So those sorts of things are huge complexities. The other thing is, and I'm not against people, companies and making profits and stuff, but we have to remember many of these companies are small companies that have very limited resources, and they may be in a position where if they, say, well, we think our drug works, but we had this side effect that we got to worry about or such. Wall Street's out there listening, and their stock price may go, which can be a huge, that can kill a trial. It can kill a program. So, they may be in the awkward position of having, on the one hand, to be positive and trying to encourage the community because they want people to participate in the file. That this may be something they've spent their life trying to develop a drug for, and yet at the same time, they have to be strike the right balance with the investment community. And that may, if the program ends up falling through, you may end up having a lot of disappointment and such. So you've got a whole bunch of different economic factors and discussions and conversations that come into play, which I think really hasn't gotten enough attention in this space. I think one of the other things that's a challenge is investigators. We don't really, we have a situation where many investigators, are like myself. They're at retirement age. They may be getting ready to retire, and so they're leaving the communities where the research is taking place. And we're not seeing enough people replace those investigators out there. And this has become a huge concern, a huge challenge, because we're essentially going to be paying for it for the next 20, 30, 40 years until we can catch up, if we can catch up. So, having people, and particularly in the rare disease community, where you need to have people that are working with these rare diseases and are trained investigators, can also be a factor. And I think we have to recognize those sorts of issues as being a concern too. So I've thrown out a whole bunch of different things. I think there's a whole bunch more that we could talk about. I do think the idea of you have to take every disease a little bit as an N of one and consider, is this something that needs extra attention as a rare disease versus, or an ultra-rare disease versus a rare disease. And that's something that I think is really going to be a challenge, because you've heard all sorts of reasons why something shouldn't just be based on the numbers, why we need to consider these other factors. And it may be that what's needed are more flexible approaches from FDA that don't even have to fit in any particular program in order to be helpful that way. But I do think that this is not going to be an easy solution, and I'm not sure that just declaring a certain level of being ultra-rare diseases, what's that going to get? What's that going to mean for conditions that are not ultra-rare but are still rare and not common? And I should disclose there that I actually have a rare disease too. I have multiple myeloma. I've been treated for it for over 10 years now. It is a rare disease. It's not one of the ultra-rare ones, so I'm lucky in that respect, I guess. But I think there's a lot of issues out there, and there's a lot of patients in all categories that are suffering. So, not an easy set of questions to answer there, and I just I think we have to really stop and think, what are we hoping to achieve with this? What's going to be the, is it going to make a real difference? Because I think that's the other question we're constantly asking ourselves. Are these programs, what do they do? Do they actually bring help that's useful, or do they have no particular benefit in the long run? Because we're talking, there's a lot of resources that are required in some of them, and we have a limited number of resources overall. So we have to figure out how to get the most bang for the buck in terms of the many diseases that are out there, the many ultra-rare diseases. So.
Well, thank you so much for taking us through all of that. This is the challenge panel, so it's okay that you went through all of the issues. That's right. We'll get to...
I didn't go through all.
Oh, that was only some. Okay. We'll get through the solutions later this afternoon, but I did want to pick up on where you just left off, which is really the topic of this event today, which is around the definition of ultra-rare versus rare. So I do want the entire panel to comment on what you just left us with, which is how should we characterize defining ultra-rare? Is it beneficial or is it not beneficial? We'll start with you, Emily.
Okay, solving the world's problems, right? I think we have to, so my bias is clear here. We are ultra rare. We can't play the game that's been defined. It's like going to play a game of baseball. We don't have 9 players or however many players there is, right? We have half a leg. And that is, like, we feel at a huge disadvantage to be able to satisfy standards that are simply impossible for us. And it's just numbers. So for us, it is a question of what is reasonable, and what can we legitimately do, which doesn't, Justin and I were talking before, like, they've been in trials for some 20 years. If I had to put all of our patients, or a meaningful number or percentage of our patients, into one trial for a decade plus, that is unethical, in my opinion. You saw the array of symptoms. Let's face it, unless it's a gene therapy, which is still, that's our only promise right now for curative therapies, unless it is that, I'm leaving people behind. And I can't, in good conscience, do that. So for ultra-rare, for me, I do see a value in defining this in a way that can be operationalized, that enables us to work with all of our patients, not just a few, and for a time frame that makes sense. I don't want to just extend a trial, for extra years to be able to properly power it. Maybe. And meanwhile, all these other products I've been working on in the preclinical setting cannot advance to a new inflection point. And that's where you also see the valley of death for a different reason, not financial, but there's a motivation factor with humans. So for ultra-rare, for me, I definitely see a value in that. I can elaborate further. I want to say one other comment, if I can. I've heard this from the prior panel, as well as the concept around regulatory flexibility. I would propose, and while God bless it exists, I'm grateful for it. I wish I could access it, but there's that. So discretion is a subjective challenge, but I wish we could think about this, and this ties into your question about regulatory appropriateness. And that, I think, reorients the conversation about what is appropriate and gives us sufficient flexibility for these different conditions. So.
Sure. So before we get to Rini, I do want to come back to you for just a moment. Are you saying that we do not need a numerical number for ultra-rare disease, if you were to summarize your point of view?
I have a point of view that's not been vetted. I think there should be a cap on the percentage of people that any given trial should recruit. Like 20%. As a PAG, we are motivated to identify all the people in this country or around the world living with a disease. That is our motivation. So if we're looking for people to enhance diagnosis, to enhance quality of care, then we know we have the best number. And if there's a certain percentage of that number that can be recruited into any given trial, that gives us an opportunity to have multiple potential trials, ideally. And so that's where I would look at it, not so much as a static number, but what percentage of the known population should any given trial have to recruit up to.
All right, that's helpful. Okay, Rini, we'll come to you.
Yeah.
What's your perspective in terms of what's the characteristic we're talking about, or do we need a numerical number and a definition?
Yeah, and I'll try to be quick. I mean, I'm coming at this from a drug development perspective, right? So what's feasible to do in a clinical trial setting? And some of that's driven by numbers, some of it's driven by the nature of the disease. If it's an incredibly fast progressing disease where everyone dies within a year, you can probably do an outcome trial in a small portion of people and see a difference. But we've talked about the complexity of many of these ultra-rare diseases which complicate that situation. It may mean that you have to focus in on certain age groups because symptom burden varies by different age groups. So I think at the end of the day, Emily and I are in the same boat in terms of Barth syndrome is always going to be ultra-rare, and the other rare diseases we're in are large enough that we felt we could do that second trial. There's some point between 10,000 and 150, where that's almost impossible. And I think it's dictated by more than patient population size. So I do think that it's really a sliding scale of regulatory flexibility based on the feasibility of doing multiple trials, and safety probably should come into play as well, right? I mean, we talk about risk versus benefit, which kind of comes after efficacy. Maybe in these ultra-rare settings, it should come alongside efficacy. You know, if your risk profile is low and the potential for benefit is there and the patient tolerance is there and the likelihood of ever getting to a conclusive efficacy determination is pretty tenuous, you know, maybe that's the shift that should be made. But I think some of the existing regulatory framework gives that flexibility. I just think there needs to be a little bit better definition around where and how aggressively to use that. And that, I think, could take into effect both size of patient population as well as some of the other factors around trial feasibility.
Justin, let's come to you.
After the first panel and those two answers, there's no bait left on the hook, I feel like, to even try and go after bait at this point. So I'm actually gonna not answer the question in that I think
Are you going to use a media trick on me?
I think it's, we're talking about this, I think, for policy development, right?
That's right.
So I think it depends on the policy that you're wanting to develop, if it's helpful or not to do it. And so if it's clinical trial design and that's a number, then I would need to look at the clinical trials and is there an area and a patient population where that seems to occur, because I don't know that number and I don't know that information. But I don't know this is just clinical trial design. I think there's a lot of other issues that are at stake here and go into play. And so, you know, is it one number for clinical trial design and another number for something else? I don't know that I can pick a point and say, yeah, that's the number, because I don't know the question we're trying to answer with that number. So I think I would need to take it on a question-by-question basis. So that's the way I'm going to back out of that.
Okay, so it depends.
It depends.
Okay, there you go.
That's better, thank you.
So AJ, yeah, I will come to you. Maybe you can talk a little bit more about the unintended consequences in those two scenarios that Justin just laid out, right? For clinical development purposes and then for policy reasons.
Well, ideally, policy should enable the clinical development, or enable at least the clinical development and the regulatory science necessary. So I like to think about it in terms of, and this kind of reflects what you were just saying. We need to include safety in this as well, because there are potential unintended consequences that we may not realize with some of these treatments. And this is a hypothetical. Don't look for any drug out there, but this is the sort of thing that I worry about when I think about these things. You know, let's say you have some sort of neuromuscular disease that's similar to muscular dystrophy. In this particular case, the children die within 2 years of being diagnosed. Terrible disease, and treatments developed that keeps them alive for like 30 years or 40 years. And you know, the short-term safety looks good. The studies are done over two or 5 years, something like that, and it looks good. But 15 years into the lives of these children, they start showing cognitive issues that kind of resemble Alzheimer's. Now what? You know, I mean, those sorts of things are potentially issues that could happen, and we just don't know at this stage. What's the right benefit risk there to consider? I mean, the fundamental issue for drug development is you want to have a favorable benefit risk, because every drug has some side effects. We don't ever say that it doesn't have any. So you want to have a favorable benefit risk. The benefits outweigh the risk. And you need to know how to use it, what the dose is, things like that. Whether you should observe certain, monitor certain labs or things like that to know about a known side effect or those things. But basically, that's what drug development is about. That's the sort of information that companies need to provide to FDA so that FDA can weigh whether or not this drug should be have its labeling approved. And that question, that fairly simple question has been the topic of, probably at this point, hundreds of thousands of meetings between FDA and sponsors, and in every drug, there's always some hitch, some complication. Nothing's ever completely clear. So that complexity is out there for anything, whether it's rare disease or common diseases. But I think rare diseases, we often just don't know as much. And what you do in drug development is you try and discharge risk. You start with not knowing a lot about the drug, and trying to evaluate this to see what is the benefit risk? How can I move this closer? And at the end, when you submit your package to the FDA, you want to be as certain as you can possibly be based on that development program that yes, there is a favorable benefit to risk, and this drug, does hopefully will help patients. And I just think that it's very hard to do that. And I think that uncertainty, we're never going to be completely, the way statistics are, keep in mind, you can have a drug that works. It's going to work in most patients most of the time, and in most of our trials, it still has a 5% chance of failing, because that's the way the statistics work. So, I think we have to recognize there may be some cases where there are drugs that don't have any benefit that get approved, because we have limited knowledge and we're afraid to put people on placebo or control or that. There may be cases where there are drugs that get approved or don't get approved where they've got some positive benefit, but we just can't detect it because of the issue. So I hate to be a realist, but that's what we deal with.
Yeah, so just quick response. The question of type 1 versus type 2 error, which you just flagged, maybe type 2 is more important in ultra-rare, because I'm not sure that you can ever discharge the type 1 risk when you're dealing with such small trials.
We started a conversation around regulatory flexibility, and we've talked about how for each of the different disease conditions, it's definitely dependent on the disease condition and the circumstances, of course, that surround that drug development process. If you were to ask for the kind of flexibility that you would want from the agency, from FDA, and the consistency and predictability of what that flexibility ought to look like, right? Do we always have to go case by case, or are there particular characteristics of that flexibility that you would want? Let's start with you, Emily.
Well, I'll do my best. With the flexibility, and I mentioned the word appropriateness, and there has to be, we talked about severity, we've talked about pediatrics versus adult. There's ways to stratify populations, and then that can inform appropriateness. I think I'm going to take this a little bit different direction. I think we have to be open to, let me say this. We're in DC, and in DC there's, not reading the headlines today or anything, but you are innocent until proven guilty beyond a doubt, shadow of a doubt, right? That's kind of just our modus operandi in this country, a belief system we have. And for ultra-rare, I'm not talking about safety, I'm talking about efficacy. You are assumed to be guilty of not working until proven without a doubt that you do work. So this concept around confirmatory evidence is really at the core of this question around regulatory flexibility. And if the numbers do not allow you to debunk without a shadow of a doubt, then there has to be a way to still advance therapies for these populations. And how do you do that? And that's where I think we have to look at creative clinical trial designs, creative data sets, and some common sense to, I know that can be challenging at times, but, and that is subjective, but to really evaluate and have in this risk-benefit framework, do the benefits likely outweigh the risks? And in some cases, that may mean, thinking through, like, what are those data sets? Mixed methods approach, like we have sociologists, ethicists in the room. the patient voice, the patient perspective. How do we systematically incorporate that into our evaluations? So for example, this trial, and I'm thinking Nike, right? Nike didn't become the brand it is today because they sold 100 shoes out of the gate. They became the brand because they had a good seller or one or two. And so natural history, you can't begin to capture all elements of a disease out of the gate. And so there always are going to be some gaps and some imperfections. In this trial, for example, one of the things we didn't check for was anurosis, bedwetting. Like, we didn't ever thought to use that as an endpoint. And yet, we saw these men get their dignity back, these children who would go to sleepovers, things that are just basic human dignity that they were able to experience for the first time while on therapy. Did we think to capture that systematically and submit that in a regulatory package? No, we did not. So that's where I do think regulatory appropriateness, when do we need to think about alternative data sets? How can we be reasonable when we find something that's really meaningful that we never anticipated?
Rini, do you want to comment on that question?
I'll try to be
Is there
quick.
some
Yeah, I'll try to be
ability for
quick.
us to predict?
Again, coming from a drug development perspective, as somebody who has to actually raise money to pay for these trials and convince investors to stay with a program for 10 years that we've been at this, and it's a really small patient population, and I have a legal background, so I'll throw that in too. I think that the construct of like what's in the guidance, what's in the law, and what does the regulatory precedent say becomes important. So while I do think at the end of the day, it does become a case-by-case decision because things are more complex than regulatory precedent, I don't think that we should ignore the precedent because investors and companies like mine make decisions on that basis. Like when we were stuck with like, how do we find a path forward when trial designs we are putting forward are not considered to be adequately powered despite global feasibility informing the size we can enroll, we turned to a retrospective natural history control style trial looking at regulatory guidance, looking at regulatory precedent, and tried to follow that closely. But if you can't really, if that's sort of not in the vocabulary that you're supposed to be referencing because everything is absolutely case by FDA, you have no roadmap. So again, investor perspective here, but that's really chilling of development. Like you need some roadmap and some framework within which you can gauge your odds, right? Gauge your risk, sort of, that's the risk benefit from a drug developer perspective. What's the likelihood I can get this right? Even assuming I'm, completely naive about the effect of my drug, but still, and that's where I think some of the black box aspect of case-by-case decision-making becomes problematic. And we do need a little bit more ability to look at least at precedent.
Yeah. And AJ, do we need those guardrails? Are there elements of those guardrails that you can define?
So, I think FDA does, issues all sorts of guidances. I do think that this is the sort of thing that it would be worth taking a look at some of the precedents out there. And, the regulatory science evolves over time. We used to only do, randomized placebo-controlled trials and that, and now we're using, there are studies that use natural history, there are, and registries, there are other sorts of approaches out there. And cell and gene therapy is raising a whole new set of potential issues because in some ways the same patient can be their own control in some circumstances. So, although it's also raising all sorts of other issues that we have to think about. So I think and FDA does try and go through and update their guidances on a regular basis. And I think, we saw the presentation, where we had all of these initiatives FDA is trying to do and trying to partner. And, I think increasingly you see things, I just saw a workshop that was announced on a particular topic today with the FDA's newsletter. And so, that's actually, in some ways, the good part of that is they're trying to respond to the evolving science, the evolving needs in that are out there. And yes, regulations and guidances take a while to be finalized, but that's a lot faster and a lot more straightforward than trying to get something through Congress right now, to be quite frank. I think we need to look at all levels of influence, and are there topics? Is there a need maybe to try and Tim, you've been involved in this with other with rare diseases where it's individual rare disease. Maybe for a group, there's a need to think about, in dealing with ultra-rare conditions, what are some approaches that might be suggested as part of a guidance from the community that FDA then could look at and could consider? So that, I think some of those things are worth thinking about.
Yeah, some hard work that needs to be done. Okay, Justin.
We're getting close to lunch, so I won't make it long. I think accelerated approval process was brought up earlier on the first panel, and I think about smaller patient populations and heterogeneity, and I think accelerated approvals in our space could really be helpful in letting things play out. If there appears to be a good signal, if the drug appears to be safe, we have some smaller companies that are trying to invest, getting those drugs approved, and then monitoring and continuing the conversation with regulatory afterward could be potentially a powerful thing to get some drugs to patients, but also continue to monitor. So I'd put a plug in for that.
Well, thank you so much for those considerations. As we close the panel, I just want to go down the line. As you talk about each of your disease conditions, AJ, as you have the broader view, what is the one lesson learned from the drug development process that you want to leave behind for this audience? It's important for really this topic of the day. How do we ensure that we can get the development over faster, for patients? And certainly we want to see the outcomes that are favorable, both on their lived experience, as they're waiting for these drugs to get over the finish line, but obviously for access as well. So what are the most important elements from what you have learned as you've gone through your drug development process that you want to leave behind?
I'll say don't let perfect get in the way of good. I've already said that once. And then instead of looking at all the reasons why not to do something, look at least one or two good reasons why to. Changing that perspective, I think, would open up a lot of doors for the ultra-rare and the rare disease community.
So unfortunately, I'm not sure I would do this again, but conversely, it's probably been one of the most rewarding experiences of my professional career. And I would say, particularly in the past 6 months, the power of emerging therapies, the power of better disease awareness for ultra-rare. We had a case study that kind of went very public, emergency access kind of case study. And since that family talked about their experience, we've received over a dozen new requests for emergency access for drug, over half of them from babies in acute cardiac distress. That's in the past 4 months. That's over 10% of the US patient population. We only thought a couple of these kids were born a year. So it's kind of real-time validation of this model that FDA's leaned into for a very long time, that you kind of have to give it a try. I mean, that's what accelerated approval is about, because that is going to improve disease awareness, diagnosis, and standard of care for patients. So at the end of the day, I'm just not sure I'd be able to raise the money to do this again, but incredibly rewarding, because I think it really does impact lives.
Yeah, I mean, I think at the end of the day, it's a complex thing to do, and it's a challenge. But I think we need to remember that there are patients at the end of this, and that the goal is ultimately to benefit patients. And when I say benefit patients, I really mean that you want that benefit to be meaningful. So you don't want to have. It doesn't help if you benefit somebody in some way and they have some sort of terrible side effect that they can't live with, because they're not going to keep taking the drug in many instances. So we need to try and get it right, but ultimately it's people that are involved and families that are involved.
I think the power of the patient voice shouldn't be lost in all of this. And I hope I highlighted the directness with which FDA in particular has reached out to try and establish dialogue. I guess you don't probably do that if everything is crystal clear and you have an easy path ahead, but they're trying to work through the problem with us. And I believe that continuing the engagement is really important. I think from the Niemann-Pick type C perspective, we've actually found it very helpful to be part of the discussions with regulators at those meetings. So if there's type B and type C meetings, being there and being part of the conversation. Everyone seems to be happy with those meetings. I don't know what they were like before because we weren't there, but they seem to be productive meetings. And I think being part of the discussion, being part of that is helpful. And the last thing I'll close with, we haven't really talked about patient-reported outcomes data in clinical trials. And I would like to think about really well-designed PROs as part of clinical trial data. Obviously, it's not probably going to be the primary endpoint, but trying to construct those things so we can incorporate the patient voice into those data packages rather than doing it at the end where we're bringing patients up before. Let's do it in a well-designed way that we can get the patient voice throughout the clinical trial experience.
Thank you so much, Justin. Everyone, please join me in thanking this panel.
Great, thank you all. Provocative comments, and I now stand between you and lunch. But it's important for us to reflect on what we've spoken about already and where we're headed or not headed, depending on the wishes of these kind of gatherings. So where have we been? Let me give you a little appetizer to chew on before your luncheon fare. We talked first about CARE being an acronym for clinical, access, regulatory, and ethical. There were those points and more touched on this morning already in the prior panels. We descended from more policy level of organizations down to individual disease entities and challenges. And I think we came out with a few things. So just something for you all to either reflect on or challenge when we get to the question and answer periods. We discussed ultra-rare being something beyond just numbers or just numbers and severity, and that there were issues with feasibility and barriers that need to be addressed further, and potentially could be by various policy aspects. We talked about the confounders. It's a small world after all, not just at Disney, but throughout the rare disease community, and the natural history heterogeneity, the endpoint validation issues, the placebo arms all confound any of these efforts. So how do we begin to address those? And that leads us to the uplifting forces. We talked about collaboration. We talked about the simulation and AI type exercises for synthetic placebo arms. And as we go forward into lunch and talk about some of these levers, I'd like you just to consider, how do we build on the current infrastructure? We heard comments about the fact that there's not as many principal investigators for some of these things, even though there's expanding need. So how do we develop the kind of ecosystem that will allow rare and ultra-rare studies to proceed expeditiously? And finally, how do we avoid type 5 error? Type 5 error is solving a problem too late, and that's what we're all struggling with. So as you make a new friend at lunch, please be back at 12:45 and we'll continue with our next panel. Thank you.
Okay, everybody, we're gonna pull it back in. We love all the networking happening. We have so many great sessions to get to. If everybody could get their coffee and their brownie, find their seat. Gonna keep talking. Everybody could find their seat. Grab your afternoon caffeine. There are delicious brownies over there. And then find your seat. And we're gonna keep going. All right, thank you.
Thank you, Annie, and very much looking forward to your considerations for challenging questions in the session following this. But I have the pleasure now of introducing our next panel. The title of it is Getting to Approval, Rare Disease Approval Success and the lessons that can inform a characterization of ultra-rare. And I'd like to introduce our panel moderator, Kishore Hari, who will self-introduce and will bring you all up. Come on, Frank.
Hello, everyone. Now that we have food in us, we can talk about successes, finally. My name's Kishore Hari. I'm a senior program manager at the Chan Zuckerberg Initiative. And it's a real honor to be here with all of you. CZI is kind of early in our rare disease journey when you think about it. We started our rare disease work just about 5 years ago. So we are still learning, like all of you. But when I see what we've done, which is really founded on the principle of not just being patient-centered, but patient-driven, because if we invest in the capacity, the organizations that patients have built, we see amazing things happening in research, in the patient communities themselves, and ultimately in the development of therapies. And what has been remarkable in the iterations of the Rare as One network that have now existed is we've seen successes come onto the marketplace. But those successes aren't just successes, they're hard-earned. They represent years of effort and sacrifice by the patients. And what you're gonna hear about in this session is a number of successes. But we're also gonna talk about the deeper layer behind those successes. What goes into them, what we should learn from them, what we should not learn from those successes, and what the future holds for all of this to go from establishing a precedent of success to a pipeline of successes across the field. I'm really honored to have an amazing panel with us today. I thought we could really quickly go down and do some introductions and then kick off with a set of presentations of 2 cases and then a set of presentations, 4 presentations, and then we'll have a longer discussion amongst us.
I'm Matthew Ellenwood. I'm the Chief Scientific Officer at the National MPS Society. By training, I am a comparative medical geneticist. I've worked in preclinical translational work in the MPS disorders since 1998 and came into advocacy in 2020.
Hi, everyone. Such a pleasure to be here. I'm Teresa Fecto, and I'm a Director of Medical Affairs at Biogen, and I lead up our U.S. medical affairs activities in ALS, which, if everyone's familiar with the space, has been largely focused into a tofersen program over the past few years, which I'll talk a bit about today.
Hi, I'm Lynn Albizo. I'm the Chief Public Policy Officer for the Immune Deficiency Foundation, of which there are over 450 different types of primary immunodeficiencies, all of which are rare or ultra-rare. And I've been with the Immune Deficiency Foundation for almost 8 years now.
Hi, pleasure to be here. My name is Kevin Thornlow. I'm Senior Medical Director at Farming Healthcare, and am in our clinical development group and oversee a number of our clinical development efforts for leniolisib, Joenja, as you may know it, for APDS, and we'll speak to APDS a bit today. Thank you.
Frank Sasanowski, I'm Vice Chair of the EveryLife Foundation, and I was at FDA a billion years ago, and in that capacity, I was asked to, and I figured out why the original Orphan Drug Act wasn't working. It was my research led to the 84 and the 85 amendments that made it work, and then I was asked to write the implementing regulations. So I've been involved for a long time, and probably about 1/3 of all the new chemical entities approved for rare diseases I've had a hand in. So today I'm just honored to be part of this panel and to be able to bring some gray hair wisdom, you know, in perspective.
Frank, you look amazing for somebody that's been working at this for a billion years. That's a say. Matthew, would you like to kick us off?
Thank you. If we could have my slide, please. Oh my gosh, can you see that? I can't see it up there. So I'll be talking today about MPS 7, mucopolysaccharidosis type 7 SLY syndrome. SLY syndrome is a unicorn whose parents were zebras. It is definitely an ultra-rare condition. We estimate there are between 100 and 200 patients worldwide. It is also a privileged zebra unicorn. The MPS disorders are all, for the most part, soluble enzyme defects, simple inborn errors of metabolism. They have well-known substrates that accumulate. These are conserved in evolutionary biology by 640 million years. The same compounds stored in avians and humans cause neurodegeneration. There is no doubt about the toxicity of these primary disease activity biomarkers. Furthermore, Emil Kakkis, who founded Ultragenyx, took this program through with learnings that he had from the approval of hyaluronidase when he was at BioMarin. So this was a very privileged program that was able to successfully get approval, and it only had 12 patients. So some of the things about this disease, as I said, it's very well understood. Simple enzymology, genetics, etc. We have extraordinary spontaneous large and murine models for this disorder. When I started in the field, the joke was there are more NIH grants to cure SLY syndrome than there are patients in the country. And it was actually probably true. And the dog and cat and mouse models have been very important. And I say models, they're homologs. It's the same gene, the same enzyme, and the same pathology. We did not need to model it in any other special way. So we have a lot of benefits in that area. All of our diseases have some heterogeneity within one indication based on how much enzyme activity is there. The serious forms almost all have neuropathic disease. And we heard from Sharon King discussing Batten's and Jason about Niemann-Pick C. The neuropathic MPS disorders follow a very simple and similar pattern of neurodegeneration and death between the age of 5 and 15. Importantly, Emil concentrated on the non-neuropathic forms in this disorder. So he was just looking at the variants of this disease that had somatic disease that could be more effectively modeled with the traditional feels, functions, and survives criteria. There are a number of regulatory issues that are difficult for these disorders, as with many other situations. And there are some things that we have already in our arsenal to help approval of rare disease: the priority pediatric disease vouchers, accelerated approval when it's used. Accelerated approval when it's not used is also a barrier, so to speak. One of the things that Emil did with this program that was incredibly valuable, he had only 12 patients in the initial phase 3 trial. And they were in what's called a delayed entry placebo-controlled structure. So every 8 weeks, a patient moved from placebo, IV administration of enzyme, to therapy. And evaluations during that change allowed individuals to be their own control group for a clear, functional readout. In this case, it was the 6-minute walk test. It's kind of crazy to me when I know how profoundly serious these diseases are that we're approving them on a 6-minute walk test. But it was quantifiable, it had been used before with MPS 1, and using this innovative approach to the structure of the clinical trial, individuals could be their own control group. We had 3 individuals entering treatment from placebo at 8 weeks, at 24 weeks, and at 16 weeks and 24 weeks. And they also used novel statistical approaches. You've all heard of ANOVA. They used ANOCA, analysis of covariance, and that's as much as the statistics as I can explain to you right now. It was really an amazing success. But what it is not, I'm not sure if my other, yeah, it's not important. What it is not neuropathic disease. It was rapidly the response was a rapid response, but it wasn't addressing the developmental disease. For the rest of our conditions, we still have the challenge of getting to the patients before they start to manifest disease, because the model for the neuropathic forms is going to be prevention, not treatment. And the difficulty in evaluating that clinically with the traditional feels, functions, and survives criteria will be cognitively. And we're looking at development in children who are 2 years and older. There are different modalities to evaluating that. There's no unitary test that patients can take, and it takes 5 years. No company is going to survive during that period. Other difficulties are thinking about biomarkers, which we have, which are excellent, and we think, especially based after our program at the Reagan-Udall Foundation, heparan sulfate as a biomarker in the CSF is an appropriate biomarker for accelerated approval. And I will remind everybody, the criteria is reasonably likely. It is not, completely defined for all time. Reasonably likely to predict clinical benefit. If we can even get an approval based on that, we still encounter other hurdles, particularly enrollment of a confirmatory trial. If we have to have enrollment by the time the marketing approval is given, the whole program is dead on arrival. Many of these programs will require newborn screening. That rollout will be 5 to 10 years to get the kinds of patients in reasonable numbers, and we're probably talking 40 patients a year for some of these conditions. So there needs to be flexibility at all levels. I think I'm a fan of the ultra-rare designation, not as a rigid criteria, but it will give review teams some kind of framework to know this is not, my grandfather's Oldsmobile, right? This is something unusual and requires a more unusual and flexible approach. Thank you.
So you gave yourself an eye chart. I'm going to try to avoid that because I'm not really going to speak to this slide. Can I switch myself, or can I get the next slide, please, for the group? So I'm going to talk a bit today. My goal today is, one, not to put you to sleep after lunch, because I am going to give a little bit of a biology lesson, but I'm going to try to take you on a little bit of a journey around tofersen and the development program. And one thing to really keep in mind, I'm going to focus most of this conversation on the past few years as we proceeded toward accelerated approval, which we received last year. But this journey really started more than 30 years ago, when Drs. Brown and Sadiq identified SOD1 as the first ALS-linked gene. And for today's conversation, really important to remember, Matt, you set me up really well. We did achieve an accelerated approval for tofersen just this past year. Quite a journey. Definitely, as my good colleague Steph Fredette would say, not all rainbows and unicorns along the way. It's been quite a journey. But that accelerated approval indeed does mean that we achieved that based on a reasonable likelihood of predicting clinical benefit of a surrogate endpoint. In our case, that is neurofilament light chain specifically, or NFL. And that also means that the clinical evidence is still being established. And we are working on, some of you in the audience who follow the ALS space, we are working on that now through our ATLAS trial, which is actually, as far as I understand, pretty unprecedented in that we're looking at the pre-symptomatic SOD1 ALS space and trying to really understand what tofersen's potential is for delaying onset of disease for folks who have that SOD1 mutation and an elevation in neurofilament light chain as a criteria for enrolling in the randomized component of the trial. So since we're going to talk a lot about the journey around neurofilament, I want to just do a little bit of level setting. So not going to give you the full biology lesson, but I do want to make sure we're all thinking on the same page here. And neurofilaments are really sort of the backbone of neurons. It kind of makes up that cytoskeleton. And when the axons of those neurons are damaged, that neurofilament leaks out where it ultimately makes its way into the blood and the CSF, where we can measure it. We can actually take some quantification of it, right, and understand what it's doing behaviorally. They're nonspecific, so it's not just in ALS that we see these neurofilaments acting, but we do know the literature is pretty well established in ALS that neurofilaments tell us something really specifically in ALS, uniquely around the progression of disease and survival. More specifically, If you have higher levels of neurofilaments in ALS, we see faster disease progression and we see shorter survival most typically. There also is a really well-established relationship here in our SOD1 literature, and we're going to talk about some of the biological plausibility here. But let's take a scan back. As I said, on this slide, you can see a lot of bullet points. I'm not going to speak to every piece on this slide. Some of it you can kind of read through and get a little bit of background. But what is SOD1 ALS? SOD1 ALS is caused by a mutation in the SOD1 or SOD1 gene, and that leads to a production of a pathological form of SOD1 protein that ultimately is toxic to motor neurons. And that degeneration and death ultimately of motor neurons is what causes the symptoms in ALS, the muscle weakness, difficulty breathing, and ultimately we know that ALS is uniformly fatal. So ALS as a whole is pretty rare. It affects what we estimate today, based on the epidemiology that we understand at this point as genetic testing is expanding, we estimate that it affects about 68,000 people worldwide as a broader condition. When we start thinking about SOD1 ALS, it gets increasingly rare. So historically, we've categorized ALS in terms of familial and sporadic, so two kind of basic categories, basically that hinge on whether or not you know if you have a family history. And we know that's a very imperfect mechanism of categorizing people. Family history knowledge, first and foremost, I don't know about you, but I definitely don't know all of that in my family. We know some things, but also we are at the mercy of when a diagnosis may have come on board in the medical community. We also are learning a ton about the genetics of ALS, and as we learn more, we're getting better in terms, the conversation around precision medicine came up earlier, but we're getting better at understanding what the genetics of this disease look like. And with that growing evidence, we're starting to really understand that the epidemiology is shifting, and about 15% or more than 15% now of ALS is considered to be genetic ALS, and we know that genetic forms of ALS can occur in people who have the apparently sporadic type of ALS. So you don't have a family history, but you've got a genetic cause of your ALS, right? So thinking a little bit about that, now more specific to SOD1, SOD1 is exceedingly rare. It's about 2% of the entire ALS population. So worldwide, that's less than 3,000 people estimated based on the numbers we have today. In the US, even smaller, 350. That's the estimate that we're working on right now. Again, as we get more genetic testing on board as standard, that number may shift over time, but it's unlikely to shift dramatically, I would say, because it's a pretty exceedingly rare condition. It's also exceedingly heterogeneous. So SOD1 alone, over the past years, as the research has evolved, it now has over 200 different causative variants of SOD1 that are associated with ALS, 200 plus. So that makes it quite difficult, challenging, and also fun to be in this space and trying to advance our understanding. The disease duration is also extremely variable across those variants. The variants can range from less than a year of survival all the way to more than 20 years survival. And the quotes around median survival time in SOD1 ALS is about 2.3 years in the current literature. So despite all of that heterogeneity and complexity, we do understand SOD1 ALS as a disease quite well, similar to what Matt was saying on the first presentation. And tofersen is really designed to kind of target that mechanism of disease in terms of the SOD1, the toxic SOD1 that attacks the motor neuron and ultimately leads to its degeneration. So in accelerated approval, if you call back on the concept of that plausibility, the reasonable expectation that we can see clinical benefit to a change in a biomarker like neurofilament in our case, that's really based on, moving away from regulatory speak, I think that's based on two key things. It's based on biological plausibility, and it's based on the evidence to support that relationship between the disease, the marker that you're looking at, and the relationship between those two things, right, and the outcome that you want to see. So tofersen is designed as an antisense oligonucleotide, or an ASO, since that's a tongue twister.
And it's really designed to target that SOD1 messenger RNA, which is really the instructions for making the protein that we were talking about. And the goal of that is to reduce the amount of SOD1 in production, that accumulation, which is total SOD1, not just the toxic, but all of it, to give the body the ability to kind of naturally clear the existing SOD1 and hopefully preserve the integrity of the motor neuron, because you don't have as much there, which should mean theoretically you don't have as much toxic protein there as well. And, okay, and so we also have the luxury, as Matt was saying, we have the luxury of a great animal model, and the preclinical work there has really shown us that neurofilament has been able to reduce axonal injury and neurodegeneration. It's been able to prolong survival in the animal, in the rodent models. So that's really wonderful progress. And the phase 1/2 work of the tofersen program has allowed us to select that 100 milligram dose that was part of the journey of the VALOR trial. If you follow this space, you know that we started enrolling that trial in March 2019. We had a total of 108 people enrolled. Move forward, that was in March 2019, move forward a couple years when we finished the trial and did the primary analysis. Accelerated approval was a success this past year, but we had a lot of learnings and challenges along the way. We missed our primary endpoint of that phase 3. So, with the way it was designed originally when we were looking at more rapidly progressing versus slower progressing, but the fact that we had an open label extension in our trial allowed us to dig deeper. So we dug deep and we really tried to understand the biomarker data, and we saw that the reductions in SOD1 showing us that target engagement, that took about 8 weeks to happen at its maximum potential. It took another about 8 weeks to see that neurofilament light level drop, so at about the 16-week mark of the study. But at 28 weeks at the end of the study, the discernible clinical the clinical benefit wasn't there from a statistical standpoint. We could see the trends, but the statistics didn't support the efficacy in terms of a primary endpoint. So we moved forward, we looked at the open label extension data, and in July 22, we filed an NDA with the FDA on the basis of an accelerated approval, and really were able to use neurofilament at that point as a baseline covariate to help us control for that substantial disease heterogeneity, to really look at it there and sort of think about now looking at who started tofersen early at the beginning of VALOR versus who started in a more delayed fashion, so they were on placebo for 6 months during that original trial, and then they started taking active tofersen at the point that they enrolled into the open label extension. So early start versus late start, and what we saw there were, we started seeing those changes, and the biomarkers were really powerful, the data were really powerful there. And we also had a causal inference model that the team worked to develop to really help us understand, essentially looking at each person as their own control from a statistical modeling standpoint, to see what is the relationship between neurofilament and disease progression. And ultimately that model showed us that at the 16-week mark, when we saw that neurofilament reduction, it was related to the 28-week mark where we saw the discernible clinical benefit starting to occur. But it took a lot of hard work and a lot of people that are far smarter in statistics than I am, and I cannot tell you the depths of those stories. But there were some challenges, and I think in the Q&A we can kind of get into some of the pieces that we really, some of the key learnings that we had in terms of where we over and underestimated the power of some of the things that we learned early on. So thank you.
We're going to tag team over here, so you can have the. So I'm going to kind of level set and sort of set it up for Kevin to talk about APDS. I'm with the Immune Deficiency Foundation, and our mission is to improve the diagnosis and quality of life for every person affected by primary immunodeficiencies. And our vision is a healthier day every day for every person with immune deficiencies. So basically, as I said earlier, primary immunodeficiencies, there's actually over 450 different types of PIs, that we say PIs. They're also known as inborn errors of immunity. And these are conditions that are genetic, that basically either the body's either part of the immune system is missing or not functioning properly. And as a result, this can cause infection, malignancy, allergies, autoimmunity, inflammation, all different kinds of issues. What we find is there's all different types of PIs, and with all of them, some of them are deadly. Many of them, if people have immune deficiencies for long periods of time, can cause organ damage and all kinds of cancers and other problems. So, so then I'm going to just talk about a little bit about some of the treatments available before we get into the APDS discussion. So over half of those with PI have common variable immune deficiencies. These are antibody deficiencies. And interestingly, immunoglobulin replacement therapy is the standard treatment for CVID, and that's been around since 1952. It was used during World War II, and then it's been used for PI since the 50s. And that's still something that we work on for, of course, it's been improved over the years, but that's still the standard treatment for those with antibody deficiencies. A lot of you may be familiar with SCID, severe combined immune deficiency. That's also been known as the boy in the bubble disease. Basically, infants with SCID will not live because they have very little immune system and once their mother's immunity is worn off, they're not going to be able to survive. But luckily, and with a lot of work and advocacy from IDF and JMF, we have newborn screening for SCID, which is wonderful. One thing I'd like to say about that is that when we're talking about prevalence, before we had universal screening for SCID, which we got all 50 states in 2018, we used to say 1 in 100,000 babies a year are born with SCID in the US. Now we know from doing universal screening that it's 1 in 50,000. So still very rare, very, very rare, but there's twice as many infants out there than we knew. So I think with rare disease, as different types of screening and identification becomes available, you're going to find out that rare and even ultra-rare diseases may be more prevalent than we thought before. And in addition, there's a subtype of SCID, ADA SCID, which was actually the first gene therapy. And anyway, so that was done in 1990. The young lady that did the first clinical trial is now in her 30s, and that's great news. And but the problem is that it still has not been approved for, and that had to do with the going to market and issues like that. So there's those challenges that we talked about earlier. There's also now recently we've had a few successes, and that'll lead me to APDS, but congenital athymia is where an infant is born without a thymus. Now there's a certain regenerative treatment that allows an infant to have an implant that allows them to generate T cells without the thymus with this other therapy. And WIN syndrome is another one that was recently approved. APDS is I'm going to pass it to Kevin so he can talk more about APDS, but that was a huge success in March of last year, and I'll let him talk about that.
Thank you, Lynn, for the summary on primary immunodeficiencies. So just to start off, as Lynn said, I'm going to provide just a little bit of an overview on our experience with Joenja that was approved in March of 2023 in APDS patients ages 12 and up. So just start a little bit with what is APDS? Well, it's activated PI3K delta syndrome. So we abbreviate it as APDS because that's a lot easier to say. And in essence, these patients have mutations in their PI3K delta enzyme that results in hyperactivity. Now, this really is a success story of science and medicine together and the community that's come together with this. This was a disease that was not identified until 2013. So just think about that. From 2013 to 2023, we as a scientific community went from identifying a disease to having an approved therapy. So really, a lot of things had to align with that, a lot of work from folks. And I'm not going to do that justice in the next couple of minutes, but we'll try to kind of summarize this a little bit. So as Lynn indicated, so APDS is one of 450, now close to 500 different types of primary immunodeficiency. And a very similar story to what you've heard from other folks that have spoke today. This is a heterogeneous, very severe disease. The prevalence of this is estimated at about 1.5 patients per million, so quite low prevalence. That would equate to somewhere around 500 patients in the US. These patients suffer from infectious issues, recurrent infections, and a number of other manifestations that affect various organs. So they have gastrointestinal disease that these patients deal with. It impacts their spleen, their liver. Chronic liver disease occurs. Failure to thrive, so short stature in these patients. Some developmental delay as well in terms of cognitive development. And very interestingly, as you think about immunodeficiencies widely, and applies here for APDS, so they have a hard time fighting off infections. They have a hard time raising an immune response to that. Instead, many of these patients raise an autoimmune response to various components of their self. So it's a very complex disease. And one of the things we heard this morning was this heterogeneity, not only in terms of manifestations, but not all patients experience all these symptoms I just described. And the symptoms don't always show up with the same severity, and they don't always show up at the same age from patient to patient. So those three things make it very difficult to put together a clinical development program, identify endpoints that can be assessed. If you think about common diseases, you can get around that a little bit with your inclusion-exclusion criteria by studying a smaller piece of the population. As you've heard today, in these ultra-rare conditions, a lot of times you're encompassing 25% of the known patients in your clinical study. So that does make it very difficult. Luckily, in this scenario, we're able to use some commonality across these patients. And I'll just touch on this quickly because I know we're quite short on time. So this program really started as a phase 2, phase 3 study. The phase 2 was 6 patients that were studied, ages 12 and up, and that was positive, and then moved to the phase 3 study that you can see here. And this is a sort of your standard placebo-controlled study, recruited approximately 30 patients, 10 in the placebo group and 20 in the treatment arm. This was done as a global study, as you can imagine, across 9 different sites, and really needed that footprint to be able to recruit this number of patients. The NIH, as one of the sites in the US, recruited the majority of these patients. They actually recruited 16 patients. And some of you may be aware, some of you may not. The NIH actually pulls patients from the United States, but also globally from other countries. And that was really important in terms of delivering this medicine to patients in ages 12 and up. Also, the ability to treat for 3 months or 12 weeks, as you see here, and being able to see changes was really important. So to sort of jump forward here and just some data, and I think the reason I'm showing the data is not to show the data to you, but just to show you how beneficial and how obvious the efficacy was here. So there were 2 different measurements as a co-primary endpoint. One was measuring changes in the B lymphocyte. So this is a type of white blood cell. And in these patients, it's well known that the development of those B cells, there's a block. They only get so far and then they stop. They can't transition to what's called the naive B cells. If you look at the graph on the right, you can see that In blue, those are the Joenja or leniolisib treated patients. You can see treating with leniolisib resulted in their naive B cells going back up into the normal range, which is the shaded range. The placebo group stayed pretty flat. So that was, kind of a hallmark of disease and very obvious result. And if you look at the p-value here, we've talked about p-values today and making sure that we get below 0.05. You can see there's a lot of zeros in that p-value. So we were very lucky and the data was very clear for us. And that also landed in terms of the second part of the co-primary endpoint, which was measuring a lymph node size. So these patients have a lymphoproliferative phenotype because of the overactivity of PI3K delta that results in a very large proliferation, abnormal proliferation of their lymphocytes. And that, of course, then leads to a lot of the autoimmune manifestations and issues that I described previously. But you're able to measure that in terms of lymph node size. And once again, very clear changes in that as a primary endpoint. What about safety, right? Safety is the thing we think about first when we think about risk-benefit. Very good safety profile we saw with leniolisib at the doses utilized in this study. I'm not going to get into the details here, but most of the adverse events that we're seeing were grade 1 or grade 2. So those are the lower adverse events, the least severe, and there were no treatment-related serious adverse events. So the efficacy was obvious, hit the primary endpoints in the phase 3 really well, and the safety profile was quite favorable. So this resulted in FDA approving the drug on review. And, part of what came out of that was clearly what I just described in terms of the data in the pivotal program, but the fact that, and I haven't mentioned this before, but the drug, Joenja, leniolisib, is actually a PI3K delta inhibitor. So we know genetically these patients have a hyperactive PI3K delta. The drug inhibits that. So the mechanism was very clear. I think that was very helpful from a regulatory perspective, along with very, very clear data. So I know we're short on time. I'll stop there. Thank you.
Thank you. Thank you very much. What I was going to do is kind of shift a little bit. When Annie Kennedy asked me to speak today, and on this panel, and these were wonderful presentations, and she said, well, you have some other experiences that might be relevant, Frank. And I says, yeah, but let me also first try to set the stage, because we've been talking all day about, do we need an ultra-rare definition, and what should that be? Well, the question is, well, why are we thinking about this issue? So I wanted to back up for a second. And the first thing I want to talk about is, what are the ways in which, the FDA is empowered to approve a therapy for a rare or ultra-rare condition? And it all depends upon showing one thing, and that is substantial evidence of effectiveness. That was in the 1962 law. And what the 1962 law said is that depends upon a showing with adequate and well-controlled studies. And so that's the key phrase. And my good friend, I can say my good friend, Bob Temple, is the one who helped then create the implementing regulations in 1971 to define that at 314.126, to say what is an adequate and well-controlled study? And you've heard many people today talk about, well, is it feasible for an ultra-rare condition to have an adequate and well-controlled study? And so that's why I want to set the stage, that the framework that the FDA has to work within has to work to this 1962 law that says everything has to meet substantial evidence of effectiveness. The original way in which the FDA interpreted that, and it still is good law, and it still is the way the FDA operates, is if you have 2 adequate and well-controlled positive studies. That's meeting the original 62 standard. There was a 1999 standard that said, Congress then said, rather than 2 adequate and well-controlled positive studies, you sometimes can have 1 adequate and well-controlled study with confirmatory evidence. You heard Dr. Maynard refer to that earlier. She was so wise, because what she said was, I don't know your exact quote, Dr. Maynard, but it was something like this. It was like, most rare disease therapies are approved on the basis of this standard. 1 adequate and well-controlled study with confirmatory evidence. You just heard Joenja. That was 1 adequate and well-controlled positive study, not 2, 1, But it had confirmatory evidence because you had other evidence that told us that there was a reason to believe the co-primary endpoint results that were positive, they weren't due to chance. Instead of having a second study show that it wasn't due to chance, you have other confirmatory evidence. So that's why those in the rare disease community have not talked enough about what Dr. Maynard was sharing with us earlier, and that is we're almost always talking about an approval that's going to be based upon not the 1962 standard, but the 1997 standard of one with confirmatory. And then the last was in 1998. Dr. Woodcock and Dr. Temple were the principal authors of the May 1998 Clinical Evidence of Effectiveness Guidance document. And we have the privilege of having Dr. Woodcock with us today. And what they said in that was, in addition to these other ways that you can get an approval, sometimes you can get it approved on the basis of a single adequate and well-controlled positive study when it's very persuasive statistically. And if that's been interpreted to mean at least a p of less than 0.01. And usually, when the finding is on irreversible morbidity or mortality, so that it's likely to be unethical or unfeasible to be able to do another trial. So those are the three ways. The important thing to note is that what I have at the bottom, you heard many people today talk about accelerated approval. What you have to remember, and even, we talked about that for tofersen, is accelerated approval. But accelerated approval requires that you still meet substantial evidence of effectiveness, which means you still have to have a showing on one of these three ways. And I'll just jump for a second to the tofersen example. Did we have that in tofersen? Did we have 2 adequate and well-controlled positive? No. Did we have 1 that was positive with confirmed? No. Did we have 1 that was highly statistically persuasive as positive? No. So what did we have? We had flexibility. So I'm going to move to that. But I just want us to understand the framework in which all of our discussion is occurring today. And so regulatory flexibility is what we saw exhibited in the approval of tofersen for SOD1. But let's look at exclusivity, because it may not be possible to have an adequate and well-controlled study for every rare disease, and especially not for ultra-rare. And I don't need to go through, because lots of people have talked about that today. But the bottom part is, since we've talked about flexibility, this regulation that I'm citing at 314.105, this regulation has been around since the mid-'80s. It's been around a long time. And what this has said is essentially the FDA is saying we can be flexible because, and that's what the FDA has done. And it was kind of, Tim Franson was kind enough to give a shout out to me. It was my paper in 2012 that kind of illuminated, how often FDA has used flexibility. That showed for the first time that 2/3 of all the FDA approvals of new chemical entities at that time, from 1983 when the Orphan Drug Act was created till June 2010, it showed that 2/3 of the time those approvals were based not upon one of those three showings, that the FDA was doing what it did for SOD1. It was being flexible. So I just want to set the standard and say that other people, and I'm not going to go into this, other people like the Europeans have found a different standard for rare diseases. That is, they've created different, and there are at least 2 people in this room who have been with me to FDA meetings, and with the senior FDA official at the table, looked across the table and said, why can't we have something, give us something like we have in Europe? Because then I'd have the ability to give you something, because we don't have these other standards that we have. I'm not going to talk about that. That's a whole nother story. Because what I did want to throw on the table today as a jump starter for conversation is this concept of maybe what we could do is look at whether or not the known and potential benefits of a therapy outweigh its known and potential risks based upon all available scientific evidence. That is, we trust the FDA officials and their judgment to be able to make judgments about the 3 standards that I talked about and the application of flexibility and whether or not something should be accelerated approval. Why shouldn't we trust the scientific judgment of the FDA to also make a determination as to whether or not the known and potential benefits outweigh the known and potential, when you look at all evidence, not limit the evidence to just evidence that comes out of an adequate and well-controlled study. But that way you can look at the mechanistic information, like with tofersen. You can look at all the exposure-effect relationships. You can look at the correlation between SOD1 protein and ALS-FRSR. You can look at the correlation between neurofilament light and ALS-FRSR. You can look at all these other things that were not pre-specified. That was not the primary endpoint. So I think, and this is not a new standard, because what I'm citing here again is a 1980s regulation from the FDA, in which the FDA said that our application of statutory standards for marketing approval recognized the need for a medical risk benefit judgment in making the final decisions on approvability, and consistent with our regulations, FDA will consider whether the benefits of the drug outweigh the known and potential risks, and you have to look and make this determination in consideration of the severity of the disease and the absence of alternative therapy. Isn't that always the case? It's always severe what we're talking about. And there's always the absence of alternative therapy. So this is empowering, and it's something for us to consider, that the FDA at the time of accelerated approval, and I had the benefit because our chairman here said, I've been around a billion years, and one of those billion years was I sat at the table at the commissioner's conference room, had the privilege as a young scribe to write down what Bob Temple and others were saying to create accelerated approval. ACT UP in the AIDS community didn't force the FDA to do this. It was not a congressional idea. It was the genius of the FDA to respond to the needs of the AIDS crisis. In the same way, the FDA can, on its own initiative, and now I'm going to go into Latin, sua sponte, on FDA's own initiative, it can basically seize upon authorities that it already has and apply it using its good scientific judgment. So I'm going to race through a couple of recent examples. Lenmeldy is a case of just 2 months ago, there was a drug approved for treating metachromatic leukodystrophy. I had the privilege of helping on this one. And what FDA says when you have natural history is that the FDA's RWE guidance says you have to come to the FDA in advance, get the FDA to approve your protocol and your SAP if you're going to do RWE, rely upon natural history. Did we do that for Orchard when I helped them this? No, we did not. The FDA exercised flexibility in overlooking the strict application of what FDA says in its guidance documents. So even though this looks like, like it fits because it had a successful outcome, it had a successful trial if you rely upon an externally controlled trial, there was still flexibility applied in it. So when people point to this or other examples, like I helped on Spinraza and Zolgensma, both for type 1 SMA, you can say that these outliers, like Lenmeldy and Zolgensma and Spinraza, are examples of where the FDA can be flexible enough, use its flexibility to get patients access to therapies, but these are the exceptions. This is not what we can't catalog for you are all the ones that have fallen away and where patients no longer have an option because investors have walked away. They've worn out with time. You heard Dr. McCarthy talk about she spent 10 years at Stealth and she don't think she can ever raise money again for that kind of therapy. So I'm pointing this out kind of as a counterexample. I'm saying FDA is wonderful, but we shouldn't get misled into thinking that this is the answer, that flexibility is enough. We need to go beyond flexibility, especially for ultra-rare conditions. 2 more examples quickly. Skyclarys is a drug we have Ron Bartek in the room. I can't believe this is like having Moses in the room, and so we have Ron Bartek in the room. Ron was the one in 1999 who created FARA, the Friedreich's Ataxia Research Alliance, and he created FACOMS, a natural history that we were able to rely upon to help my client Riata. I helped them for 10 years. Get this drug approved for the first drug for any ataxia that was just approved last year. It was really on Rare Disease Day, February 28th, 2000. I thank Dr. Baracchio for picking that day to approve it on it, but it was just beautiful. And then another example is Duvyzat that just also got approved about 2 months ago for Duchenne's muscular dystrophy. In both the cases, Skyclarys and Duvyzat, you had one adequate and well-controlled study that hit its primary endpoint, It wasn't highly persuasive. It wasn't less than 0.01. But you had confirmatory evidence. The natural history comparisons provided part of the confirmatory evidence. And I'm saying we have shining examples where the FDA has given access to patients who are suffering to new therapies like Lenmeldi, like Skyclarys, like Duvacet. And we've heard plenty of other examples. Look at the Joenja, Tofersen, Mepsevi. I don't think you said the name. Mepsevi. On all these drugs, these are shining examples of what the FDA can do by extending flexibility. But what I wanted to challenge us as a community of people who are dedicated to the rare disease cause, I want to challenge us to think about whether or not it's really unfair to expect the FDA to always apply flexibility. Like under what circumstances, how far can I stretch flexibility? It's not clear, it's not laid out anywhere. And so instead of what we do is we come up and give the FDA and say, FDA, look at science has changed since 1962. We have translational science. We can look at SOD1 protein. We can look at genes. We can look at, let's evolve. Science has evolved. Let's evolve our regulatory standards. You have these standards, they're there. Instead, look at whether or not the known and potential benefits outweigh the known and potential risks using all scientifically available information. So that's what I wanted to bring to you today. My closing slide is that when I had the privilege on Sunday night, 60 Minutes did an interview with Pope Francis. I want to say that when I met with Pope Francis a few years ago, I asked him for a blessing on all of those of us who have rare diseases and all of us who help those with rare diseases. He gave me that blessing. And as a devout Catholic, I want to tell you, that means that each of you has been blessed. So I just want to pass that on. Thank you.
Thank you so much to all of our panelists. I know we started with successes, but Frank moved us into potential solutions here, and I'm going to get to that in 1 second. I want to start with our 3 case studies really quickly. I want you to rewind the clock for us. There are a lot of choices embedded in each of your stories. Knowing what you know now, would you approach the problem differently? And then I'm going to have a part 2.
Do you want me to start? Okay, sure.
Anyone can start.
Yeah, so a little piece of the story that I didn't have time to mention, but that phase 3 study took 6 years to happen. So that's, that obviously is a long time. But it was successful, at least, right? That's one component. The other component is, and we've heard that this discussion today about, kind of the commercial opportunity and the investment up front. And so this drug, leniolisib, was originally with Novartis. They discovered it. They started the work in APDS, and Pharming brought it in as an in-licensed drug a number of years ago when the phase 3 study was part of the way through. So we finished that off and put the regulatory file together and achieved the approval. But my point there is that this was something that was a small indication that for a larger company, many times isn't attractive or prioritized in their portfolio. And a big part of being successful with something like this, obviously we've talked about the hurdles of a development program, and those are not easy. But then you have to be, successfully commercially. And how do you do that in such a small number of patients? And I think it takes a very special organization. I think it takes a very devoted organization. I think Novartis sort of saw in Pharming its experience in rare disease. So our other commercial product is Ruconest for hereditary angioedema. It's a drug that we had originally was licensed to somebody else and didn't go so well because of some mergers and whatnot. And Pharming actually brought the drug back in-house and commercialized it themselves. And it's been a success. So I think Novartis saw that, the devotion that a small company with a very good focus could deliver. And I'm not trying to sort of brag about farming, but what I'm trying to get to on this really is that there really has to be that focus and emphasis on education. Most people don't know what APDS is. I know you're probably not surprised by that, but they don't. Most allergists and immunologists that you would talk to in standard practice don't know. So it's really important as a company and as a community that education gets out there. And with that awareness, then they're thinking, oh, this sort of sounds like my patient might be an APDS patient, perhaps. Let's do the genetic testing and see. So there's that awareness side of things. And patient finding for clinical trials, one of the sites that was originally put together with Novartis was actually in Russia. Now, this was 6 years ago. The political landscape was a little bit different. But then the patient finding efforts that are really required to make the drug a success and get it to the patients after the fact, after approval. So I think that's it's not all sort of rainbows and low p-values as I showed you. So
Rainbows and low p-values.
And
the next talk.
If I can just add to that, when, particularly with education and finding patients, patient advocacy organizations are really essential. And I know with the Immune Deficiency Foundation, when we worked with farming, we started doing lunch and learns. We had, educational. We have a web page on APDS. Like, we worked with them to help educate the patient community, because there's many people like that may be misdiagnosed with CVID and other types of PI that that they can go and talk to their immunologist and find out, maybe I should have genetic testing and find out if this is something. So the role of patient organizations is really important in this journey.
And I think, the question of will we do it again, really, it's an interesting thing to learn about through the tofersen journey. And, we've learned a lot through this program. And I think two of the key learnings, one is we've talked a lot today about heterogeneity of disease. And we know that ALS, SOD1 or otherwise, it progresses very non-linearly, so there can be rapid progression followed by periods of stabilization and kind of going on that roller coaster ride for an individual. And that really challenges the ability of traditional measures like our, what is still considered a relative gold standard in the ALS space of the ALS functional rating scale, the ALS FRSR, as you hear a tongue twister said repeatedly. And we understand that the ability of a measure like that, which was our primary endpoint, is kind of challenged on those reasons. And we now know that neurofilament did a much better job of predicting prognosis or disease progression in our study than the ALS FRSR did. But at the time the study was designed, it was designed based on what we knew, right? And that has evolved over time. And I've seen a very simple slide that my colleague Steffordette has created. And it's a very simple overview of the neurofilament literature that we understand. And if you think about a timeline left to right with publications listed, over the past 15, 20 years, that literature has expanded very, very rapidly. And based on what we had at the time, I think the study was designed in a pretty meaningful way. But now I think we have the ability to integrate things like neurofilament into covariate framework in terms of understanding how to more effectively control for disease heterogeneity at baseline. And also, the do's and don'ts. I think we had some excitement around very early learnings in the tofersen program that were before my time at Biogen, but I've learned a little bit about that. And I think we and the community sort of overestimated some things and we underestimated some things. There was a lot of excitement about the early work, but I think where we underestimated that I didn't really have time to go into is the maximum biological activity. By reducing SOD1 and reducing that neurofilament level, it took longer than we expected to reach that maximum activity. It also took longer than we expected for that maximum activity to translate to clinical benefit. So all of that preceded clinical benefit, and it took much longer than we thought. So for tofersen, we learned that 6 months wasn't enough for a trial based on the way tofersen works, but that may not be true of every product. So.
I'm in the very deeply uncomfortable position to speak for Emil Kakas, who developed Mepsevii. I think he may not well have gone through with it. It was the cost for such a rare indication is extremely high. And I think the most important thing that is learned from the lessons of the MPS community is the consistency of staffing and communication from the review teams. And that's borne out clearly. Emil's written about it in his book documenting the development of luronidase for MPS 1. He had the go-ahead to use urinary gags as a surrogate endpoint. And they came back with the unfortunate death of the head of the review team, and they changed their position. They needed a placebo-controlled trial, and they moved forward with that at great expense, so that now BioMarin does not market luronidase. They had to give it over to Genzyme, changed the trajectory of that company fundamentally. So we really need to make sure that we have access to these accelerated approval designations. They're not good for all of the inborn errors of metabolism. Some of these disorders do not have a clear-cut soluble biomarker. For many of them, though, it's duck soup. And making sure that we get that message across, I'm not sure how we do it, but also making sure that the staffing and the consistency of the guidance from the review team at the FDA is important, because one of the things we do hear consistently is every time they meet the review team, the faces have all changed, or many of them have changed, and the goalposts are shifting. And in a rare disease indication, I get that. It's difficult to bring new people on and get them up to speed, but I'm hopeful that ultra-rare designation will give a certain level of oversight and also flexibility and empowerment to review teams to make difficult decisions.
I think we have time for a couple more questions. And I'm going to bite on the provocation that Frank threw out there. Would the new statutory definition that Frank threw out, would that have changed anything for the development in your 3 cases? Would that have helped? Would it have hindered? Anyone concerned?
Well, just to, if this mic, is this
Yeah.
on? It just to make sure, just so you understand the question, because it's a great question. And just, so if we had, just to restate it, if instead of the standard being you have to hit this p-value, that you have to, or Mepsevii, with these 12 subjects, instead of looking at it from the prism of how do I get an adequate and well-controlled study, in front of the FDA that's going to have a positive result so I can meet the standard of substantial evidence effect. Instead, can we look at all available information that's scientifically sound, instead looking at a risk-benefit, looking of do the known and potential benefits outweigh the known and potential risks? So that I'm just restating your question.
Yeah, I agree. I think, in these indications where it's very difficult to measure the harder clinical endpoints because of the small sample size that's available to recruit, or the duration of treatment that might be needed, I agree fully, Frank, that I think it really comes down to the risk-benefit, and that the FDA, regulatory authorities being able to use their flexibility to assess that. What's the unmet need in the population? What are the risks of the therapy? And what are the benefits? And really nice to see that flexibility being used for different biomarkers, as we talked about today, for these different programs that are biomarkers of disease and speak to the potential for benefit.
Yeah, I mean, it's a great point, and is this on? It sounds a little different to me. I think from a biomarker perspective, also, we have the luxury now of knowing a lot more around neurofilament, and that can begin already to inform trial design in ALS and potentially other spaces.
But neurofilament light for us as our surrogate endpoint is really meaningful in terms of how our mechanism of action of drug works. And every drug may not move neurofilament. And I think we have to be really thoughtful also in terms of leaving room for therapies that work in different ways. So I think that's a caution. But also I think from a study length perspective, we're fortunate to have had the benefit of the flexibility with the accelerated approval pathway, because 6 months simply wasn't enough. And if the tofersen program had been stopped because of that original failure, obviously there would have been a very different outcome. And it's costly. It's costly to do these things. So I think there are many considerations, and I think the increasing flexibility, and Frank, to your point, I think coming, as we all say to folks that we manage and businesses that we're all in, come to me with solutions, not only problems. And I think what Frank is suggesting is thinking about opportunities for the field, for this community, to really define next steps or support the defining of next steps in offering some viable solutions.
Did you want to weigh in?
I love Frank's characterization. That's not a world many of our programs experience with the FDA, and I wish it was. But even today, we have problems with control groups. When a control group for a neurodegenerative disease, I have trouble with them existing at all. We know that these diseases are uniformly fatal. How many dead children do you need to power your study? I mean, in my mind, that's what it comes down to. That's very stark and an unpleasant imagery, but that's it. One study we know of, children were allowed in the placebo arm, it's called the rescue arm, to lose 25% of their developmental quotient. They will never recover. They will experience an early death and likely psychosis along with it. We have a program that is in work now where there's a standard of care, where there is an anticipated fatality from bone marrow transplant of 15 to 5%. Why does this exist when the treatment modality being evaluated does not have a fatality associated with it? This is madness to me. And I don't know how we figure out to get FDA to come to creative solutions in these situations. These are one-offs, but we should be able to fix this.
One final question. Approvals isn't the end. There's the next that comes after that. There's more successes to be had. How do you think about now going from approval to getting the right treatment to the right patient at the right time?
I will start in that. I love that terminology from Sharon King. We work very actively within newborn screening. For many of our diseases, if we do not get in pre-symptomatically, we will not see an adequate response. The period of nomination is going to be 2 years after we get an FDA approval. That's a best case scenario. And then the rollout nationally by state testing labs will probably add another 5 years to that. We're looking at 7 years before we can reliably recruit enough patients. Getting studies pre-enrolled for a confirmatory trial, that is a recipe for a program that's dead on arrival. So we need to figure these things out. Access is going to be an issue. A lot of these gene therapies, there may only be 6 centers in the country. And with the number of children we have on Medicaid, we need to get that fixed. Some of these things span so many departments within Health and Human Services. We need a coordinating interagency committee on rare disease that is talking to HRSA, CDC, NIH, Social Security, both NCATS and NICHD, to figure these things out proactively.
Quick reactions, because we're just about at time.
I'll echo that, and I mentioned the Atlas study. Pre-symptomatic is the focus there. I think there's a good statement, but also just
beyond the clinical trial space, I mean, we're all talking about identification, and one of the things we've been focused very heavily on, and our team, is getting the education out around genetic counseling and testing and ALS. We have the 23 guidelines that were published. We finally have an actual document we can reference, but getting that implemented and understanding what's needed in the way of implementation science, so we have some relative standardization to improve access equitably across the community, is really important.
Yeah, and I would add, and you mentioned all the different departments under HHS. I mean, FDA approval does not necessarily mean getting treatment to the patients. Most of the time, and the work that I work on is access. It's coverage for different kinds of treatments. And, you know, we've just seen it with Alzheimer's, which is not a rare disease, but there was an approval for treatment for Alzheimer's, but CMS is not inclined to cover that for Medicare, Medicaid. So, you know, regulatory framework does not end with the FDA. CMS has a big part of that, as well as the private insurers. If we want to get different treatments out to the, you know, to be effective to the people that need it, we need to look at the whole system of regulatory framework and allow, you know, advocate for that kind of access.
Please join me in giving a big hand to our panel.
Thank you all. Great job. And you can continue to participate because now is the open question period. And we're going to ask folks to grab the microphone. I won't presume to have the answers and may call on others who've already spoken, but just a couple thoughts to lead us into this period. You know, we've all been drinking from a fire hose of knowledge as well as concerns, and that's made life very interesting. There's also been some gaps that have been brought up in our outside discussions about talking more about N of 1, N of 10, N of 30 type trials, about several other things, and one which just came up. How do we expedite things as much as possible without getting out too far over our skis? And there's certainly documents we can learn from that help us not do that. There was a publication by FDA more than several years ago about successful phase 2 studies that didn't make the cut in phase 3. How do we learn more from things like that so that we don't overstep? So it's that kind of critical balance. Patients are waiting and they're suffering, and type 5 errors are horrible, but how do we be able to adjust for that? And then I would just ask you, as you speak at a microphone, which we have some we can pass around, choose a theme which you want to speak to, and then elaborate on your point if you have suggestions or commentaries, and starting it out.
Hi, Kevin Fryer from Salem Oaks. The reason I'm involved in rare disease is partway through my career, I was getting tired, got onto a rare disease project, and I said, this is intellectually challenging. This is tough. Everything I know about drug development, out the window. And I think Matthew Ellenwood just said it. This is unusual. It demands an unusual approach. We've talked about how the system breaks down for these ultra-rare diseases on many levels. And we've talked about what's the number we should go for. I don't think the number is the right thing. But can we characterize in a multifactorial analysis, where does the system start breaking down as the numbers get smaller and smaller? Can we look at FDA's records, or can they look at it and see where do the CRLs pop up? Where do they really struggle? Because we can't all see that. Where can we see payers quit paying for things? Where can we see access becomes a problem? All the way around, let's put all those dimensions together, probably take some sort of AI tool to put it together and say, here's your inflection points. And if we do that, and we can characterize the breakdown points, How do we create a system that does work for these diseases?
Thanks, Kevin. Very interesting. It also calls back to mind, I apologize for not remembering which panelist brought this up, but the notion was if detection and screening continue to modify our epidemiologic observations, then are we at some risk of, if we set a ceiling now and find out the epidemiology changes, you've cut out a bunch of what had been ultra-rare, if that is agreed. So just anticipating if we pick up one end of the stick, the other comes with it. These lights are really bright, so I cannot see anybody.
Hi.
Hello.
Hi, I'm Nina with the Janssen's Foundation. Thank you all for participating today. It was a really informative discussion. I did have 2 questions. The first one is I really appreciated this last panel for sharing quite a lot of the successes and some of the struggles. So as an ultra-rare disease that affects less than 30 people, so Janssen's is also pretty near to a clinical trial. My question is there a formal process or way in which we can do more of sharing of what went wrong, and where can we go to find that out? Because most of the ultra-rare diseases are run by either a parent or a patient or a caregiver, and you know, there's a lot of privilege in this room to be here, to be able to engage. But what about those who can't come because of their disease or because they're caregiving, right? So where do we actually go to find this information? Because there was really some important stuff shared today. That's my first question. And the second question is to the representatives from the FDA. My question is, some of the things that were shared in that slide about, you know, how an approval takes place and what are you looking for in terms of, you know, the 1998 law. Are there sessions that you do with patients, groups that can sort of walk through that, especially much earlier on, and not like, you know, I've heard 6 years in one phase and some of them have been going 10 years. We don't have that kind of time. So how can we speed those things up, and is there a platform for doing that? Thank you.
Thank you for the question. There may be others who want to comment as well. I don't think rare diseases have the corner on lack of data sharing across multiple constituencies. And we're starting to see more of that through several types of organizations. Critical Path Institute has been brought up, and they have consortia on things like lysosomal storage disease, the Duchenne Regulatory Science Consortium, who all are working towards, for example, pulling together information so that everyone has common access to the most current data, and it can help the planning of multiple companies, academic groups, and so forth. So how do we break down some of the barriers between what have been kind of provincial views of data? That said, I know FDA is under particular constraints about not sharing proprietary information. So we don't have the luxury of being able to ask that of them. What we need to do is to have more opportunity to sit down in groups like CPATH or Foundation for NIH and so forth to be able to foster that kind of data sharing in a lockbox type fashion, I think it's called, so that the proprietary information is not compromised. Did anyone else have commentary on this particular point they'd like to make? Kevin does, and then.
So in that mythical new system that I talked about, what if FDA could share when we're talking about ultra-rare? It's already pretty sparse there, and that could be like a kind of an exception. And we could have other exceptions too. I mean, it's that kind of radical thinking we need to get to that world where ultra-rare patients will be really cared for.
Yeah. Being a physician, I'd like to call for a lawyer on this. I'm sure there's some elements there that we'd benefit from learning from, but it's a great idea. How do we make it free and clear? You're speaking for the FDA version of WikiLeaks, I guess, but yes.
Hello, my name is Anke Arnold from Asafaros in Switzerland, a small biotech company developing drugs in lysosomal storage disorders. And I have a question actually regarding the differences between zebra and cedar. I'm not sure if it's only me who sees those differences or if it's actually true, but it seems that flexibility is handled different between the divisions. So I would love to hear more about this and your thinking.
Well, I'm not sure I'm the right party to address that, but I can certainly say that there are some significant differences in product profiles and vaccines and certain biological elements that reside in the Center for Biologics in differing from some of the, well, both therapeutic biologics, small molecules, and other elements. The CMC and so forth is very different, and we have a world authority here who can address that. Dr. Woodcock?
I can't speak for the FDA. I don't work there anymore, but you got to look at the size of the treatment effect and the mechanistic plausibility. And I think if you sort those things out, not totally, because some of the things that were alluded to about, say, the soluble biomarkers, I have issues with some of that, with some of the way that's been handled. But in general, a lot of the differences that you see in quote flexibility, which I'm going to talk about in the next panel, come from those differences, which I think Tim was alluding to. For example, gene therapy frequently has a curative effect. Okay, so you pick up your bed and walk kind of thing. So you really don't need statisticians. The statisticians would disagree with that vehemently, but no, you don't need statisticians, okay? But, if you're talking about a smaller treatment effect, then pretty soon it gets more difficult to interpret, especially with a heterogeneous disease, and especially if the disease progresses slowly. So, it depends really on a lot of the factors, but I think that it underlies some of the perceived differences.
Thank you very much.
Hello, my name is Nelmi Askilua. I'm the founder and CEO of Folia Health, and I have a brother with CVID, so thank you for the call out on his condition. I'm just curious for the group to discuss or just think more about this idea of the benefit-risk ratio that Frank brought up, and whether that's something that actually could be well served with the inclusion of more of these within patient change analyses or use of the patient as their own control group, where when you see that switch, the ability to ascertain benefit risk on the individual level in that kind of N of 1 model could be something that makes the analysis a bit simpler, a bit easier to understand where that effectiveness may be versus the risk that person experiences on their own course of the disease. So just a comment on that. Thank you.
Yeah, thanks. It's very interesting. I think there are a number of programs in various industry groups as well as at the FDA looking at unique endpoint adjudication, and how there are qualification processes that will allow new measures to be considered. So I think all those things are possible, and we heard in Dr. Maynard's comments earlier today about some of the endpoint programs that are ongoing that I would encourage sponsors to take advantage of the opportunity to speak to the respective FDA review divisions and explore those. And Frank has a comment.
Yeah, I was just going to say, since it was kind of based upon that benefit risk, what I want to say is that a lot of times, like you brought up the baseline control, could that be applied? We heard examples, Teresa talked about with SOD1, with ALS, you have relapsing remitting. People go through honeymoon periods. I used an example of DMD, Duchenne's muscular dystrophy. If you just look at age, there is a honeymoon period, like boys, I think between like 5 and 7. I don't know if I have the exact ages right. Paul Melmeyer would know, but I don't. But somewhere along there, they actually do better because even though they're losing dystrophin at a linear rate, their rate of growth makes up more than exceeds, the loss of dystrophin. So they actually have an improvement. So if you looked at a baseline control, you'd see somebody who's doing worse actually doing better on some clinical outcome. So what I'm saying is, it's not so simple as just say, but what I was arguing for is that I trust the FDA's scientific judgment. And if we empower the FDA to kind of say, look, you can look at all available information. You can look at all the mechanistic information, all the natural history information. You can look at everything. And make a judgment then on the basis of, are the known and potential benefits outweighing the known and potential risks? Then you can take advantage of all the scientifically valid information that a scientist has available to them today. And so that was my premise, is let's broaden the field, because right now the legal standard, and I am a lawyer, you were asking for a lawyer.
I did.
So the legal standard right now is the only thing the FDA can really look at is data that comes from an adequate and well-controlled trial. And so all this other information, is not within their purview, strictly speaking, unless they exercise some extraordinary flexibility. So I was saying, let's broaden their spectrum, their ability to take into and account for all available science, because science has evolved since 1962. We know a lot more. Let's allow the FDA to be able to take advantage of learnings that we have.
Thank you. Oh, right back here. Thanks, Frank. And yes, we have several questions. Annie, were you?
Actually, this isn't a question. This is more further comment on the benefit risk piece. And I just want people to understand, benefit risk is not some magical term that now has suddenly come on stage. Ever since physicians have been treating patients, we've had benefit risk. Because when you give any medication, you give any sort of treatment, do any sort of treatment, whether it's surgery, medicine, whatever, you're making a judgment about, does this do more good for the patient than it does harm? And fundamentally, that has been out there and in the both the scientific literature, but also really in a sense of the medical literature. It was mentioned that in Europe, benefit risk has been around for a while. And in fact, I remember way back when I was medical director globally for a treatment that I was responsible for, there were questions that came from Europe about, because of some adverse events that had come up, about the benefit-risk balance, and was it still favorable for the drug? We did a submission, laid out the case, and it was determined that the drug was still beneficial, more beneficial than harmful, and therefore that it should stay on the market. And that type of thing is inherent in medicine. Now, I think it's gotten a lot of additional attention from the regulatory standpoint for the legal reasons that Frank laid out. But fundamentally, this is still something, this is a basic consideration for any physician treating any patient. And ultimately, the question for the FDA when they approve a drug is, is this drug more beneficial than it is harmful?
Yeah, thanks, AJ. And I think it also calls into question how we look at benefit risk in a structured sense. And there are at least 4 different major systems that look at structured benefit risk. Pharma has an articulated program for that. EMA has a variety of formulas for that, not a variety, just several. But as you begin to look at all of these, trying to understand if they all will yield the same outcome is important to understand for patients as well as practitioners. Somebody else had a question over here. Anna, you had it.
Just a comment. Just several times throughout the day, we've heard remarks that, I'll turn around because my back's to the room, that I feel like makes me maybe a call to action to the room or just an overall comment, that I feel like it's incumbent on all of us to, as we're thinking about small populations and ensuring that what's working in broader rare or more common rare is enhanced and protected, and then we think about how we enable pathways for the smaller populations, we have to work harder to give cover to the entities that are making decisions, for rare. So I think as we're talking about, and Frank gave a presentation on how laws were passed and then flexibilities were reinterpreted, we've had regulators here today and leaders from federal agencies who we regard as heroes in rare disease, but you shouldn't have to be a hero to make scientific regulatory decisions. And I worry about some of the outcries that we're seeing in media and some of the now secondary adjudication processes that we see in the access environment that question the regulatory review processes that will undermine any discussion we're having here today around ultra, but then also are continuing to undermine what's happening in rare. And I think that it's incumbent on all of us to make sure that people understand what is currently working about rare disease as we think about needing additional pathways or characterizations. We have to make sure that we're protecting those pathways, that we're protecting our regulators and federal agencies, and that we're ensuring that we are a mighty voice for our community. As much as we're pushing for change, we have to protect what's working. And so we were just talking a little bit on the panel a minute ago about could there be additional flexibilities, and we'll talk about that more. But we have to make sure that when we say that, we're not asking for rules to be bent, because we in the rare community know we want things that work, and we want things that work better than the known natural history. And that's incredibly important. And so when something is passed and then something is approved, we have to get to every payer and talk to them about why this product works better than the known natural history. And we can't leave it up to the media to get that story out there ahead of us. So I just want to put that out to the room because that message goes beyond ultra-rare, but if we're going to start to move a message forward around ultra or small populations, we have to make sure we give that a runway ahead of that message.
Yeah, I think the communication, great. Dr.
Yeah,
Wood?
I second that, Annie, and I think the community will all be most successful if we're all together rather than trying to point fingers at one entity or another is causing the problem, but sort of collectively acknowledge that we have a problem and we need a solution. And what was just said about the benefit risk, I'd like to clarify. I think what Frank is talking about is what the evidence the agency is allowed to consider when it considers benefit risk. So it's always for FDA, it's always, and I'm not speaking for FDA, it's always a benefit risk analysis at the end of the day. However, the question is, what evidence can be relied upon to make that sort of formally, and how do you square that with the statutory standard?
Yeah, so how do we expand the intake? So, we have just a couple minutes left. Be happy to entertain comments. Or is everybody so overwhelmed with all the data and thought processes? It's been stimulating. That sounds wonderful. So we will take a break a couple minutes early. We need you to be back by 2:45 promptly. We have a very special panel of distinguished colleagues and excited to have that. And then we promise closing on time. So take
Tim, really
15.
quickly, we're going to put up one more poll for everyone to sort of think about during this break. So if you can respond to it, we can come back to it during the next session.
Are you going to put that up right now?
Yes, I am.
Okay, hang on just a second. Here we go. Survey says,
We made the QR code bigger, so should be easier this time. I am.
okay, can you see this all?
Cool. So the question is, what are the primary therapy development challenges your rare disease? Oh, sorry. I'm going to skip to the next one. Sorry. What characteristics should inform a definition of ultra-rare diseases if one were to be created? And you can select all that apply. You do not have to pick just one. So feel free to think about this on your break and answer a question.
Super.
Thank you.
And that's the one and only?
That's it, just the one. Just the
Great.
one.
So go out and enjoy coffee, and we'll see you in about 15.
Yeah, I think so.
Okay. Hello, everybody. All right, we are particularly excited about, well, we've been excited about all the panels. I'm really excited about this panel in particular to get to have a discussion with three of my personal heroes, and I'm sure many of you feel the same. I am going to do introductions of these three. I know we haven't been doing them all day, but then they can introduce themselves as well. First, I'll introduce myself. So I'm Annie Kennedy, and I serve as our Chief of Policy, Advocacy, and Patient Engagement for the EveryLife Foundation. I will also share that ultra-rare diseases is particularly important to me in that one of my first introductions to the rare disease space was when I was a senior in college, my spring year of college in April of my senior year. I received a phone call from my mother who called to let me know that my dear friend and longtime boyfriend had just been diagnosed with a rare cancer, a small cell carcinoma, and went on to learn that he was only the third, we didn't know to call it an ultra-rare disease, but he was only the third person in the world to have been diagnosed with this form of cancer. And at the time, he had been given a life expectancy of 3 months, and fortunately, he lived longer than 3 months, but over the next 13 months, which ended up being his life expectancy, my life trajectory changed significantly. I ultimately did not go immediately to graduate school as I had planned. I became one of his full-time caregivers, and I learned a lot about what it was to be a young person in a care system that was not equipped for people who were 22 and 23. I learned a lot about clinical trial eligibility and what it meant to fall outside of that. And then I also learned a lot about hospice care. And so I actually hadn't even thought much about that as we were planning for this conference, except the anniversary of his death is on every year on May 13th. So he died on May 13th, 1997. And as we were celebrating that the other day, it occurred to me that that was an ultra-rare disease. So this is a particularly poignant topic to me, as that was my first introduction into the rare disease community. So I thank everybody for their commitment to solving not just for rare diseases, but for those who are being left behind. As we have all of these wonderful wins in rare disease, we do have communities who are subpopulations of our population who are not yet benefiting from what we are learning. And so I know everybody here knows that very personally, and so do I. And so we are going to have a really wonderful conversation here with three heroes who are really helping pave the pathway for our rare disease community. Immediately to my left is Dr. Peter Marks, who is our director of the Center for Biologic Evaluation and Research at CBER at the FDA. And then to his left is Dr. Joni Rutter, who is the director for the National Center for Advancing Translational Sciences, or NCATS, at NIH. And then to their left is Dr. Janet Woodcock, who probably the only thing she has ever failed is retirement, who recently retired from FDA. And in her time at FDA, I think over 20 years, served as the director of CDER, and at many points served as the principal deputy commissioner, and then for some time also served as the acting FDA commissioner and held many roles. And we're delighted to have her back here in her not so retired role from the agency, but as an expert in rare diseases. So before we jump into what I'm sure is going to be actually a really stimulating, provocative discussion, and I also am grateful to them for coming without much prep, no slides, and no advanced questions. So they're just going to come and have a really great discussion. We did do the survey, and I know we've shared some of the information from the survey that we sent to our patient advocacy organization partners ahead of this conference. And Jamie shared this slide with you previously, asking people, what does rare mean to you? And we got a lot of great feedback. But we also asked a few other questions. So we asked organizations what they felt a formal definition of, whether a formal definition should be created, and overwhelmingly people felt that ultra-rare should be defined. And then the people on stage are never going to see this, but it was asked, we asked a little bit about their communities. So we asked people to talk about, as a patient advocacy organization, who they represented. And then we asked whether or not their communities they represented had an FDA-approved therapy. So we just wanted a little more context about who the people being surveyed were. And you'll see that a fair number of them have FDA-approved therapies. And then we asked whether there were therapy development programs underway within their communities. And an overwhelming number of them said yes. Okay, this one, the people on stage are not going to be able to read. Just for your awareness, this is like old school microfiche over there. So this question is, all right, asked about the primary therapy development challenges in rare disease. And so the first one, just in order of the most significant, are too few patients. The second, categorically, there's sort of a bucket, is a lack of outcome measures, insufficient lack of natural history studies, and diagnostic barriers. And then the third category would be study and design challenges. And the fourth, a lack of endpoints or biomarkers. And then this one, why do you feel a formal definition of ultra-rare should or should not be created? And the top answer was around the need for different incentives, which has come up a lot today. We've talked about, especially at the first panel. The second was around the support and underserved population. And then there was a tie at third around receiving targeted additional funding and concerns over ramifications of separating ultra-rare. All right, and then the last that we're going to do is what characteristics should inform a definition of ultra-rare. And we're going to talk about this a little bit today. And we've actually had some really great suggestions that have come up throughout the day today. But again, sort of bucketing some of these responses, the top being a measure of patients, the second, disease progression, the third, age, and then the fourth bucket was treatment factors, clinical trial factors, and diagnostic odyssey. And then just sort of pairing that with some of the things we heard today in our discussion, we're gonna not go right to the closing yet. I'm gonna leave it up on this one. Was throughout the discussion, just to remind people, we talked a lot about the characteristics that could inform a definition, would be defining in a way that could be operational. Could there be sort of a cap on the percentage of patients in the known population in any given trial? Disease severity, and could we be thinking about, are we actually thinking about a number, or are we thinking about more of an algorithm? So, okay, that's to maybe get us kicked off. So I'm gonna stop talking so we can actually hear from the people you guys came to actually hear speak. And maybe kick it off with a question for everybody maybe to start off with. So maybe the first is, as we think about who is it that we're talking about when we think about our smaller populations, or who maybe is currently being underserved when we think about our therapy development programs. I remember listening many times, Dr. Woodcock, to you present to our patient communities around sort of the recipe for developing therapy development programs and building registries and natural history studies, almost to the point where it was like, here was the recipe for success that we could think about. And I'm struck by, as we were watching the presentations today in the panels, and we started the morning really hearing about some of the I don't want to call them failures, but some of the communities that have not had successes yet, and how that's impacted those communities. And then we had a panel this afternoon of successes. And I'm struck by the fact that the ingredients for those trials and those programs don't look that different. And so can you talk a little bit about, and I'll open it up to everybody, how you think about that and how we think about that as we think about what that recipe is overall in rare, and maybe whether or not we need a different recipe for ultra rare, and what some of those differences might look like.
Well, I'll be happy to start. First of all, everyone should keep in mind that for regular diseases that are very common, only about 1 in 10 of products that enter a development program actually succeed to get onto the market, and even then some aren't commercially successful. So this isn't a high success enterprise, it's a high risk enterprise, it's a high failure enterprise in general, and then it becomes complicated with the factors that everybody talked about, the small populations, the heterogeneity, the inability to predict progression, the lack of outcome measures that are validated in that disease, the lack of biomarkers, maybe not having mechanistic understanding, and so forth and so on. So it isn't surprising that there would be a lot of failures. And even in regular drug development, the adage is fail as quickly as possible so you can move on to the next one. But you only have one chance maybe as a company in a rare disease. And if you fail at that development program, you're out of the game. And also it takes a long time because of the slow enrollment, the small population, and so forth. So what we were offering 10 years ago or whatever was, what can help? Okay, so development of biomarkers, development of specific patient-reported outcomes, right? Having natural history studies. Remember, we just obsessed on that, and the communities picked that up. And what we heard was that has provided success. Okay, in the sense of external controls. But it also has provided understanding, I think, of the heterogeneity, even of progression rates. We had huge arguments with the ALS community over this because they wanted to model progression, and they were thinking it was monotonic, and it isn't. And it's sort of, saltatory or whatever, and, happens in spurts. And so that creates more churn in your trial and makes it even harder to accomplish a statistical result. So, overall, in the sense it isn't that surprising, as one of the panelists said earlier, it's just very hard in the rare. It's very, very hard. The more biological understanding we have, the better off we are. And as I said earlier, we have gene therapy or something that aims to restore something. It's very mechanistically clear. You probably, I don't know, Peter, if you have the stats yet, but I think the success rate is going to be higher.
And so the initial look from the New Digs group says that, and this is all comers, so I think it's probably better than this, but at least their first analysis that the threefold higher rate of success for genetically targeted therapies, and that's all genetically targeted therapies, not just gene therapies.
Right. Yeah. But again, you're lower down on the pathogenic scale, closer to the actual origin of the problem. And so you would think, instead of trying to, modify a network later on in the process, you're trying to do a fundamental change. And so that probably will pan out, but still that means it's just 1 in 3 or 4. So it's still, and for a rare disease program, that could be, like for a whole disease, that could be a death knell to fail once. So that's part of why it's hard, I think.
I'll just say that some of the things that we're doing at NCATS are we have the way of thinking about this, the approach that we like to take is to not look at it a one disease at a time way, but to understand exactly what those pathways are in a set of diseases that have overlapping symptoms, for example, or underlying disease pathological mechanisms. And so through the Rare Disease Clinical Research Network, this is entirely their whole practice. We have over 20 of these that we support across the country, and And they bring the different diseases to the table because of that overlap that they have. So that more than one disease at a time way, I think, is one way that we can try to help raise the boats, if you will, to get at more of those diseases. So if there's a success in one pathway, perhaps in that set of diseases that they're looking at, that might be, give them a lot of rationale for pursuing other approaches that might target maybe a different aspect of that pathological mechanism or systems biological approach. So I think that's one of those things. And then I'm sure we're going to talk about gene therapy too, but we are thinking about it in the same way in terms of gene therapy with a program that Peter and I are involved in called the Bespoke Gene Therapy Consortium. And there are a few other programs that we have in this way, but really thinking about how to use the idea that there are rare diseases that are caused by a gene that cause that disease, and there might be different mutations in that gene that cause the disease, affecting different severities, for example. But if we think about gene replacement, we're really looking at the whole class of that particular gene and disease and how it causes those different diseases. And so the therapy development, when we're looking at replacing a gene as one approach, it offers us the ability, again, to really think more about how we do that in delivery, not necessarily by disease, but how to affect that in terms of delivery. And if we can solve that problem of delivery, then that opens the doors then for us to really make headway into many other areas. And so one of the things that I always talk about is if you go to the post office and you get those boxes in the post office, it's a flat rate shipping. If it fits, it ships. And so that's kind of how we're thinking about the delivery approach for gene therapy. Right. If it fits in the delivery system, then we have the potential to ship it into cells. And so this is one of those ways in which we can also attack, I think, multiple diseases at a time. So I think there are lots of approaches out there on the more research translational side that we're working on.
Can I just go back to your comment about NCAT's approach is to look at multiple diseases at one time. When you do that, are you looking for commonalities around those diseases? And can you talk about, would, for example, small population diseases sort of fit together in that model? So can you talk about that a little bit?
Yes, it's a great question. So there are largely overlapping symptoms of those diseases. So for example, porphyrias. These are overlapping diseases and caused by different genes that cause those diseases. Now, some of them have a little bit more prevalence than others. Mm-hmm. And so they're not all the definition of a rare disease. Some of them are I guess would fit under the rubric of ultra-rare, but most of it is really just looking at that overlapping symptomatology.
So lumping versus
splitting, yes.
And that rubric that we're all making up together, by the way, right?
Yeah. And it's a rubric that's worth, I think, thinking about, but it's difficult. I mean, when we're talking about the U.S., globally it's even more fraught with a variety of different definitions. So I think there also needs to be a larger thinking around this, because when it does come down to rare diseases, we want to find as many people in the world to help us get to therapies. Mm-hmm. And if we all have different definitions, which we do right now, for just rare diseases, then that might be a focus that we could be thinking about as well.
Yeah, I mean, just to redirect the conversation a little bit, I mean, and this comes from the vantage point of thinking about where we're coming from gene therapies and from talking with many in the field. I mean, one of the challenges in ultra-rare diseases is not so much just getting through the clinical trials, because we actually have ways to deal with that. When you have gene therapies that really work, 10 patients can get you across the finish line. The real issue is commercial viability of these products, which is becoming an increasing headache, because there are a number of gene therapies that have made it across the finish line, particularly in Europe, where they're available because we know that they work, but they're not commercially available. And so figuring out how to address this commercial viability issue is the other piece that I think we're going to need here to how we're dealing with the clinical development pieces. It's one of the reasons why we're very interested in seeing manufacturing technologies grow and improve, because that's going to be key. It may be less of an issue 10 or 15 years from now when we move to genome editing as the way we address a lot of gene therapy, because there it may be that you just have a machine that just you dial in what you need and it produces it. But right now, we're in a place where getting towards figuring out how to have a commercially viable enterprise is important, because without that, we're not getting products to patients.
Yeah, and that's something I've worked on my whole career, is how do you improve the success rate? How do you decrease the cost of making these therapies available? These two things are linked. Now, gene therapy, I think, is succeeding in improving the success rate, but it's still so expensive to manufacture that things like the bespoke consortium and everything are necessary, because you can't have it cost tens of millions of dollars for every single intervention you're going to do. We need some kind of plug and play intervention till we get to the gene editing place.
And I think, too, picking up on this idea of the unmet need of commercializing these, when we talk about in the translational space, we're talking about de-risking, and the really high risk is in the earlier phases of just identifying, is that target real? Is it affecting some change? There is just such high risk there, and that's not something that's going to be commercially viable of any interest whatsoever. So I think that's an area where we have an opportunity to work very hard in identifying those targets for rare diseases and try to move those forward in that pipeline. But then as we go into, along the pipeline of getting into IND enabling studies and first in human phase 1 through phase 3, the further you go along that path, the higher the opportunity for that commercialization potential. But as Peter says, this is, right now, the environment's very difficult. And so I think this is an area that is, we don't have foresight into what's going to happen into that. But this is an area that we need to hopefully support a little bit more.
So how do we do a better job of de-risking that?
The commercialization potential?
The commercialization, or even further upstream, as you were talking about.
The AMP consortium that Francis started is trying to do that, and the large companies, I have to admit, I kind of started the fad for all these consortia long ago, because I think we have to share data. We have to stop, keeping it private. And they are sharing work on, but they're looking at large diseases because that's what's attractive, okay? But they're trying to get the translational science nailed down better, so that when you have that target, it isn't just some individual researcher's brainstorm. It's a consensus scientific finding that people say, here are the mechanisms by which this disease works. I think it's no accident that a lot of the rare diseases where we've had success, they've had clear mechanistic understanding of what's going on. It hasn't been empirical, we'll just try 10 different things and see what works, right? There's a genetic basis for the disease. It's deficiency disease, or it's too much activation.
Yeah, I mean, for us, I think we can try to help from a regulatory perspective clarify some of the buckets, because the truth is that rare disease, as far as I'm concerned, that are with a genetic basis, come in a couple of flavors. They come in the really easy flavor, which is that there's a simple deficiency of a protein. The gene therapy replaces that, and you can then measure either the gene therapy. Right, exactly. Very, very simple. And it's usually, these are cases where something's missing, you fill what's empty, and then something works. I mean, that's the easy case. There's a middle bucket where, well, you can't measure necessarily what you're replacing, but you can measure something upstream or downstream, or find some intermediate clinical endpoint that's easy to measure. That's a middle bucket. And then there's the hard case where either you can't measure it, or it's a multifaceted pathway where the gene therapy is doing one thing among others. Those are the hard nuts to crack. But the truth is, there are so many in the two easier buckets, and we can try to help, I think, regulatory certainty of outlining how to move forward in these more straightforward cases may be very helpful to the community, because it'll be more straightforward from the genetic perspective,
And from the
where to go.
investor
For the
perspective.
investor, it's spot on, Janet.
Yep.
Spot on, Janet. That's what, because again, in our system, we have to have there has to be a way towards commercial viability.
pathway.
Yeah, and that's why you sometimes hear about why I'm kind of on a kick towards automated manufacturing and more and better manufacturing technologies, because if we could bring down the complexity and cost of manufacture, you could imagine that manufacturers would find it attractive to have a portfolio of rare disease therapies that could be commercially viable and could be sustaining moving forward.
Yeah, and I'll just add too, in the other, the sort of non-gene therapy side, I think the Rare Disease Clinical Research Network is a really strong place where the research can happen, because they bring together, there are 20 different consortia, but they study over almost 200 rare diseases, and some of them are very rare, and I think they're really the innovation engine that kind of helps us understand the biological pathways and those targets, and they're very focused efforts to be able to do that. And so the strength of that brings the advocacy groups together, it brings the researchers together, and ultimately we now have 12 different FDA-approved therapies through that program, which is quite impressive. So I think that is a system that works. Earlier in the phase, we're also looking at a clinical trial readiness kind of program where we support researchers to help strengthen or evaluate biomarkers for then more regulatory development. And so there are a couple of ways in which NCATS is supporting the research to help lay the foundation for driving.
How well do the other institutes play on this? Because part of what's so striking about NIH is the extreme fragmentation, which frankly the patient advocacy groups have driven. But your point is that baseline biology is biology.
Yeah, biology is biology, yeah. Yeah, so NCATS doesn't do anything alone. We work with all of the institutes and centers. And so with the Rare Disease Clinical Research Network, for example, there are 10 other ICs that help support us.
Oh, that's good.
And that is incredibly important, because we just aren't resourced to do enough. And so the more we can get that support from other institutes and centers is really fantastic. So we really rely on
They're rare dental diseases and they're rare
Exactly.
cardiac diseases. So
Exactly.
it makes
And
sense.
we are the translational people, but we need the subject matter experts to
Right.
really make sure
Basic.
that moves forward. So that institute mission-specific activity is important.
That actually is a nice segue. I enjoy co-moderating. This is great.
Oh, sorry.
No, this is great. I have asked like one question 15 minutes later. I'm like, I'll ask another. You guys are amazing. Can I just ask about data sharing? And I'd love to hear, I feel like I could just say the word and you guys will just go. But I feel like in ultra rare, data sharing is even more important than in any other space. So I'd just be interested in your thoughts around data sharing. And I'd be interested in, are there enough incentives to share data in our small population?
Who would give you the incentives? The university incentives are don't share your data so you can publish, right?
So do we need to change the incentives for data sharing?
Well, we've been talking about that for a very long time. I know David Feigenbaum, when he talked about his own disease and he looked into it, he found there were like 5 centers around the world. Each of it, they had their own little stuff. They weren't connected. He talks very eloquently about that. He was in a position, being a medical researcher, to change that for his disease. But I do think that obtains, I think things like the network change that, okay? But for companies, my experience is, when we've talked to, because I've been, harping on this for 20 years, okay? When we've talked to Congress about this and so forth, they give money to CPATH or have us give money to CPATH and so forth. But, companies generally view this as competitive or they just oppose it on principle. So that's my thought about it. I think there's a great advantage to data sharing, and most of it isn't that competitive. However, in the ultra-rare, how many people are working on it? It's mainly the researchers that ought to share data, frankly.
Well, you have PIs, you have patient groups collecting data and have registries, you have right, you have data in many different silos. So do we have the right incentives in place to be pooling this data and bringing it together?
I think the incentives that we have are just made. We could always use more incentives to share data. We see, I'm a geneticist by training, and growing up in the genetics field when I did, they really ate the young because they didn't share data. But then there was this big meeting and this big report that came out, and all of a sudden that we were all just really going for the genome sequence, and you had to share data to make it successful. And it turns out that there were more publications that happened after that than there were before, and people thought, oh, okay, well, maybe this is actually better for us. But
did the journals make that happen, or did the NIH force it?
I think
or
it
all
was
the
more
above?
NIH required more of the data sharing process. Probably journals too, though.
As I recall, they also put things in place that you had to have the back data and be able to share it and
That's
so
right.
forth.
Make it available.
Yeah.
So I think that's an important piece, is more incentives are needed. I will say, too, that that's easy for me to say. It's probably a different answer from the FDA perspective. But one of the things that we are doing as a government researcher in studying, for example, one of the other programs that we have in gene therapy is called PAVE-GT, and it's paving the way for gene therapies. That's kind of the way we like to describe it. But it is, because we're doing it as the federal government, we are able to make the rare pediatric disease indications, the approvals for those templates available to the community. We have published a paper on those templates so that others can learn what was in our template for approval in the FDA package, and then hopefully we will continue to support these. We've only had one to date because that's where we are, but we plan on publishing more of these so that others can learn from our successes and our failures. There will be failures, but our mission is really to make that available for people. So that kind of data sharing probably wasn't what you were talking about, but I do think that
great.
information and data is super important. And for the Rare Disease Clinical Research Network, we are bringing those data together in a data sharing environment to make it available to the community. So that's another way.
Maybe one of the other things when I think about data sharing, this is maybe a weird sharp left turn, but a couple things that came up on the panels today were talking about known patients versus what we then learn about patients as either products are approved, or Rini gave an example earlier today about, I think it was a compassion program, where then once that story was known, additional patients were learned of. And I'm also struck by, then we have this strange self-fulfilling prophecy in our rare space, which is then compounded and ultra-rare around the eligibility for a condition to be added to the federal newborn screening panel, and needing to have an intervention, which is often interpreted as an FDA-approved therapy, but we often can't recruit for trials until we can identify patients young enough. And so I'm just interested in, to what degree do we have the right incentives to even know of patients? We have limitations in ICD codes, which is something I know NCATS has been working on in your zebra triggers and your diagnostics. So I'm just wondering if there's anything any of you want to comment on around, is there more we could be doing to benefit our research efforts that you work with in our smaller populations, as we think about who we even know of who's living with our ultra-rare diseases or our small populations?
Well, I'm working on a project, I won't talk about it anymore, but to help patients get better diagnoses faster. Because I do think that the model, even when I was med school, like I'm not the kind of, I can't remember numbers and stuff like that. I just can't remember them. And, you're supposed to remember like the journal page where, some article was written on them there. This system is really bad, okay? And now, nobody's going to remember all the syndromes. So you go to urgent care or the emergency room or your local MD, they aren't going to be thinking of a rare syndrome, or even your pediatrician, for example. So I think we need additional ways to arrive at diagnoses of people so that they can be diagnosed faster. And we need to empower the patients to do that, not just the healthcare system.
I mean, some of this is just, to be honest, some of it is actually doing things we can do now before we do things we can do later. Like, I don't know, I mean, it seems to me like even just, we're starting to approve gene therapies, right? And yet, to get some of those gene therapies to be screened for in the United States is basically hand-to-hand combat from different states. I mean, it's the equivalent, because there are 60 some of them have compacts that they do it together, but there are 60 jurisdictions, because between the 50 states and territories, that have to decide to add them to the newborn screening list. It's not it's not done federally, right? I mean, they get added in one spot, and then that is a decision made, there's a federal decision made, but then individual states then have to adopt it, and that takes time. So just even thinking about doing what we can do, we're not taking full advantage of that. And you can see what UK is doing now with whole genome sequencing on newborns. So there's an incredible extremes here of what is possible, and where we'll come out. It'd be interesting to see.
And certainly the work that we're doing supports this idea of doing more whole genome sequencing, and perhaps
we'll learn a lot of other things too, additional things.
Exactly.
I had something I want to say before this panel's over, so just so. Now?
Go wherever.
All right.
Just keep going.
Yeah. Well, I want to reflect on what Frank Sasanowski was talking about on a prior panel, all right? The 1962 amendments that he alluded to the Food, Drug, and Cosmetic Act, called for adequate and well-controlled studies, including clinical studies, as Frank showed, And that was a triumph of evidence-based medicine. And it was hard fought for the next 20 years, And there was a lot of judicial actions and, lawsuits and so forth. So the FDA was in a position that it had to fight for clinical trials, because at that time, the alternative was anecdotes. I looked at the approval of Premarin, and they had 2 series. As one doctor in Germany gave 24 women Premarin in postmenopause, and they felt really good. They really liked it. That was the efficacy data in the file, along with a doctor in Mexico. So, and that was every woman almost goes through this at some point. It was by no means a rare disorder. So it was a triumph, and FDA it was hand-to-hand combat for 20 years to get these kind of trials done. And so it was established 2 adequate and well-controlled clinical studies, and then a p-value is somewhat arbitrarily chosen at the 0.05 level as a yes-no, because the FDA has to make a yes-no decision, Now, statistically, that doesn't make a huge amount of sense, but you do have to make a yes-no decision, So in 1962, don't forget, we didn't even know there was a genome, more or less, And so, and even the common diseases, we really had no idea of their biology. So what was put in place was an empirical testing standard, where you cast the dice, and then you did a body count, or whatever, And then statistically, you said, does it look good? And mechanistically, no, because most of the mechanistic rationales that were given to the agency were frankly BS at that time, because we didn't know enough, Fast forward 50 years of NIH investment in science, all right? And so you've seen some softening of that standard, and Frank presented it, but still, as I said earlier, the evidence that you're supposed to base this on is actually, and I don't totally agree with Frank, I don't think FDA has the discretion that he was, practically speaking, that he was saying, fast forward, we've had accelerated approval, we've had regulatory flexibility, we've had one trial and confirmatory evidence, but basically at the end of the day, the evidentiary standard is supposed to be that standard, adequate and well-controlled trials. This puts, for very rare diseases, for all the reasons we've talked about, okay, in a very difficult position, right? Because we may have very good mechanistic information, may have reasonable biomarkers, but they aren't validated, and we may not even be able to show, because we don't have enough patients and so forth, their relationship. We may have a very good story and we have dying children, right? We may have a very good story about why we should we should make that product available to those people, but the reviewers are put in a terrible position. That they're told they have to uphold the standard. That's the role of the FDA, is to enforce the law. At the end of the day, it's a scientific law enforcement agency, and these therapies have to meet the standard to get on the market. And frankly, people who develop larger diseases, therapies, they come in with all kinds of stories about why it was okay that the p-value is 0.06 and that there are trends and this and that. And so, how do you enforce a standard here and not enforce a standard there? And so what we've seen is a lot of cognitive dissonance internally, of people who are being asked to, at one hand, enforce the standards, on the other hand, be flexible. You could be flexible, and then pretty soon you're going to break. So I think we need either overtly, I think it'd be best to have a statutory pathway for diseases where there's very strong mechanistic understanding and the totality of the evidence enables a reasonable prediction of benefit over risk, or it's possible FDA could do that by regulation. Now the agency, and Peter, I don't expect you to comment, the agency can't comment on this and probably could not support such an idea, because there's a risk, I think the risk of opening a statute, there's a risk that people Annie, you already alluded to, I think, okay, there's a threat to the orphan drug incentives. Now, don't forget, the Orphan Drug Act was passed because there was an economic barrier, right, to developing. There isn't as much now because, there's a different kind of economic barrier, but back then, when you were wandering in the wilderness of biology, who was going to do that, right? And so that offers economic incentives. What we're talking about here is a pragmatic or practical barrier you simply can't do the kind of studies that were envisioned in those 20 years after 1962. Now, if you happen to be a gene therapy and you happen to be curative and you're substituting for a deficiency, then bingo, okay, you won the lottery and okay. But they aren't all going to be that way. So that's what I think.
Yeah, I just want to pick up, though, on something that you said, because I worry less about our authorities, because I think we have tremendous flexibility that we can use, at least in my area. I actually am more focused on the cognitive dissonance that you're talking about, because it's very challenging that I think many people are brought into the FDA culture, and the FDA culture, I think what my first impression of the FDA when I came was I would go into people's offices, and the first thing you'd notice is they'd have multiple years of the Code of Federal Regulations, which is about 8 volumes for us. That goes from goes from the ones to about the 1200s, and that focus, it makes it very challenging, because you come into the organization and it's somewhat defenders of the regs and defenders of the p-value.
And it's the 20 years of triumph against anecdote.
So it's one of the reasons why, one of the things we're trying to do in our center is the regs are important, we got to follow them, but start to focus on the patient, right? Mm-hmm. It is, because ultimately the regs, let's just think about it, right? The regs exist. Not because you have a beautiful reg. Like, it is true, the expanded access regulations, they're very beautiful. They're very beautiful. But that said, who cares, right? It's what you can do for a given patient. And so I think it really is changing this focus. And it seems, I know, it sounds crazy, right? People obviously know they're dealing with patients, but no, it's very easy with this cognitive dissonance to become the defender of the p-value.
Correct.
Of 0.05.
Well, because you're
Right.
enforcing what you consider
Right.
the legal standard.
Right.
And so on one hand, we have, the threat to the Orphan Drug Act. On the other hand, we have the threat to access through reimbursement, which you referred to, Annie. So, if you view something as a, quote, lower standard, then that's an excuse not to
But I think we
paying
have
for
people that are smart enough that we can focus on the patient and actually still stay on the right side of the regs. And I think it's just a matter of, in some cases, just changing how we think about things and how we approach them. I mean, I'm not saying that we don't need progress. I'm not disagreeing with you on that, but I'm just saying.
Well, I mean, that's
we have-
fair because sticking with the current standard avoids some of these other threats, right? However, my assessment is, and of course I'm outside the agency, so now I can say this, right? My assessment is it's kind of a bridge too far for some people, and there is a great deal of cognitive dissonance within the agency over this flexibility issue.
Yeah. The advantage of trying to stay with our current framework is that my great concern about changing our standard is that right now we're already seeing, I mean, I know this in my area, we have insurance companies fighting against giving kids gene therapies that are potentially life-changing because their cost and, oh, well, accelerated approval, does it really mean it works or not? No, it means that it's approved, right? But we're already seeing that now, and I worry, I don't want to see us in a place where we have haves and have-nots in this country, which I already am concerned that we may see with Medicaid and gene therapies for sickle cell disease, just to call that out. Kind of worried about how that's going to roll out, but you can imagine it happening on a broader scale if we change a standard.
I agree. However, I don't think, I don't think there should be a numerical definition of ultra-rare. I do think instead of changing the standard, having a slightly different pathway where it is not feasible to do adequate and well-controlled clinical studies and being very overt about that, whether it's by regulation or by statute, could be finessed. I think it takes some time, but I do think, because don't forget, it isn't like Alzheimer's where there's going to be like 1 million people. You're going to have to pay for this, right? Or something like that. So I just wanted to get that out there because I do think that's what we're seeing. We heard many people on these panels calling for more regulatory flexibility, okay? And I just feel like that some of the agency reviewers, they're bent backwards about as far as they can go.
Yeah, I think the other problem that we're seeing with human health is something that, it's funny, it didn't affect it when Francis started genome sequencing, right? It used to cost for a base pair, per base pair, it costs $1 a base, it cost an incredible fortune, like several dollars per base pair for sequencing.
Not anymore.
Now it's less than pennies per base pair. And so the problem is somebody had to have the courage to spend that money up front to have this progress. And unfortunately, where I see us right now is we're at this expensive portion of gene therapies, for instance, and until we actually get more into this, we're not going to see the economies of scale. We're not going to see people really interested in making fabrication devices, that is manufacturing equipment, for this that would dramatically bring down the cost because we just don't have enough, there's just not enough throughput. And so I think it's kind of a
chicken and egg
phenomena, exactly.
Yeah, and my look at, I remember I was there as a regulator when French Anderson tried the first gene therapy. Look how long that's taken. And I think the next technological steps, although they are going to be accelerated, and you all may help accelerate them, okay, it's going to take some time. It's not going to be next year where we have a factory that can spew out, inserts and delivery systems on demand.
Yeah, and I think we're trying to think about that ahead of time. Now, we're doing adeno-associated viral gene therapy delivery now, But we know full well that what we're doing now is not going to be probably what we're doing in 5 to 10 years. Is going to be different delivery systems. So we have to be thinking about the manufacturing on that. But certainly what we understand and how to do that delivery, where and how that delivery works, and then the regulatory processes that we need to do, we're going to learn a lot about those along the way just by looking at AAV. And then when we start to transition into other delivery modalities, we can be ready for that on sort of that sort of platform. And so I hope that what even we're doing today isn't going to be obsolete for, even though our delivery might be obsolete, what we're doing is not going to be.
I mean, it turns out AVV, we started with that as this is, gosh, it's like 2017, we started conversations about this platform approach. And at that point, to be honest, CRISPR-Cas9 was still kind of a twinkle and now, you have, AAV is a platform, but, the second and third generation CRISPR constructs, which are prime editors, are going to be the epitome of platforms where you're going to have something that's almost exactly alike. It's going to be a genome editor going to the liver, where it will be different by 50 to 200 nucleotides in the.
But this would be something that the U.S. could invest in. I've been trying for continuous manufacturing for the last 20 years, and I tried to get the instrument manufacturers to modify so they could have online technologies. It's been a struggle every single, but really, if this, instead of having just this company and that company doing this, if they were a huge push to automate production of this type of technology, it would be a giant boon for the United States, I think.
Agree.
Totally.
So I think that's something, and that would help people of all kind of diseases. I mean, even say hypercholesterolemia, they're the people, a lot of it's genetic. Used to blame everybody for what they ate, but actually a lot of it's genetic, right? So, it's plausible that people with severe cases of common diseases actually have rare mutations that could be better managed by some kind of alterations. But that has to make it cheaper. So anyway.
Oh my gosh, I was hoping you forgot we were all here. And you guys are just going to keep going. This is amazing. Well, I did want to ask you, this really is amazing, this conversation. I was not actually kidding. But I do want to ask, you keep talking about regulatory flexibility. Throughout the day, we've been talking a lot about regulatory flexibility. And a couple suggestions have either come up or come to mind. And one of them is, I think one of the things that as a community, everyone would really benefit from is learning from the precedent of what's gone before. Ultimately, we would learn from everything that's gone before, not just the successes, but also things that haven't worked as well, which I understand is more of a challenge. But is there an opportunity for us to create a knowledge management system that would be public and mineable, that would not just be internal to one agency, but that the community could then benefit from and understand? Some of us have been trying to collect some of that data, and we are developing case studies on our own, but is there an opportunity for a public-private partnership to start to collect some of that and understand what is working? And I think that becomes even more important, not just in rare, but in ultra-rare. So I'd be interested in your thoughts around that.
So there's some areas that are pretty easy to do that in, I think, where it's not proprietary information, but where, for instance, it's endpoints related. And this goes back to something, when I first came in, there was this question, I think, going back to, one of the challenges is people see us at CBER take one approach to an endpoint and CDER take a different approach. Now, let's just start to level set. In some cases, it's just because of bad communication between the two centers. That's something that we just have to fix, okay? Okay, let's put that aside. There are, though, cases where you're going to have to take a different approach to a biomarker when it's a small molecule versus a genetic therapy. And what I'm saying about that is that we do a very bad job of communicating to the community why, because I think if we can't put it in simple terms why we do it differently in CBER than they do in CDER or vice versa, and could put that on a website, then we don't understand it ourselves, and probably we should fix the problem
Well,
internally
I think
again.
there's a lot of that going down.
Right, right. But I do think we could probably help with this.
Yeah.
And some more transparency into how we come to our, again, and that doesn't involve letting any proprietary information out. It's just endpoints and how we handle them. And then I do think we probably should be encouraging, to the greatest extent possible, our sponsors to put stuff in the public domain, because that would really help. Small developers of small gene therapies who have succeeded would be really helpful to have their success stories get out there more. There is some insight into that from the summary basis of regulatory approvals that we post, but we probably could, again, be better at essentially explaining how we got there.
Also,
and Rick Pazder's done a good job of that in oncology sometimes.
I agree.
Right,
I think the failures are even more salutary.
Yes.
Yeah, and so that would require, that's really a private effort, that the agencies can't put that out, but it may well be that folks would be willing from a private group.
Yeah, I think, there's an opportunity with artificial intelligence and machine learning, and more pharma companies are using these approaches to understand how to look at perhaps toxicity in molecules, or why drugs are failing. That information would be incredibly valuable. But how do we get that so that it doesn't affect the proprietary nature of their work? I think if we can use those types of analyses to bring that a little bit at a higher level, we could gain a lot, I think, from that. But I think thinking about it as a public-private partnership might actually be the way to work through what that would do. And we're going to need the lawyers to help us think that through.
Yeah, well, CPATH, which Tim is very familiar with, has worked a lot of this out, and they've done pooled analyses of Alzheimer's trials and all kinds of different programs pulled together, and you do learn a lot of stuff. You can learn natural history from trial data, okay, because people have followed the patients and so forth. So I think that could be very valuable. Could a group put it together?
So, and the natural history brings me to the next topic. So one of the things that we hear, and I see you. I see you. But one of the things we hear a lot from some of our ultra-rare, all of our rare communities, but especially some of our smaller populations, is that natural history is very difficult, especially in those very small populations, and especially those very rapidly progressing diseases. And so while developing natural history studies is certainly something communities strive to do and are working to do, what is the alternative in the meantime for trials for communities that are looking to solve for something within a decade and are looking to perhaps use real-world evidence or use registries in the interim to really help bring data forward? We were spending a lot of time talking about using natural history as a comparator, which is amazing. But for some communities, they're very interested in what else can be brought forward as well and invested in.
Well, there have been drugs approved based on comparison to registry progression. I think same in biologics, right? Yes. Depends on how good the registry is, right? And have they collected the relevant outcome measures that you can put comparison, and do you have enough baseline information on the patients that you understand they're relatively comparable, and so on. So I think really rapidly progressive diseases might be somewhat easier to study.
Yeah.
Uh-huh.
I mean, the beauty is that for gene therapies, there are 2 or 3 ways to get there. You can get there if you have an animal animal models are not always perfect, but an animal model can sometimes get you there faster than gathering natural history. Mm-hmm. You can get there sometimes because you have the natural human experiment. You have somebody, it could be a parent or a relative, that has a reduced amount of a given protein, yet doesn't have any observable effect. So if you know what your target is for a gene therapy, again, you have something reasonably likely to predict. And then the fact is, we expect a lot from gene therapies. So some of these are good enough that if you can show change from baseline, right? You don't need a natural history study, because if someone goes from being able to move a limb to being able to move a limb, well, that's good enough. So you don't need to know the natural history of the disease the same way. That's an extreme example, but I think, to be honest, we have to expect a lot of our gene therapies because nobody's going to want to pay for them unless they're actually able to do something significant. So it may be that that's part of what helps get us over this. So if you understand, but this, again, it's unique in this genetic space of gene therapy and some of the genetically targeted therapies where you can see these big effects. Unfortunately, nothing that I love small molecules. They're wonderful. They're wonderful. The problem
They
is
don't work as well.
They don't work as well, and it's hard to it's just hard to predict. Even the ones you rationally design do what the heck they want sometimes, and they have off-target effects and have toxicities. Whereas it's a little bit easier with gene therapies.
And I'll add to maybe a third approach to get there, which is building off of building off of the animal models are hard. Animal models are hard, and also not all rare diseases are going to have a good animal model. One of the things that we're starting at NCATS is to build a program that is taking primary cells or induced pluripotent stem cells from patients with rare diseases and building the sort of organ system model with them so that we can use them to not just study the progression of the disease in vitro in a very small environment, but also to think about looking at potential therapeutics like gene therapy, for example. And if, for one program that we have on this is a musculoskeletal disease, and we've created an organoid system within the lab that doesn't contract, but when we add the gene therapy to it, it starts to contract. And so this is one way that might help us start to generate that evidence. There's still a lot of work to do, I think, in sort of showing that proof of principle, but that's another potential area.
But it's very important work because it also may be very important for us in the genome editing world because you'd much rather have, it may sound crazy, but having a non-human primate experiment is nothing compared to having a human organ on a chip because you can actually use the human gene, the relevant sequence in editors. So
Okay, well, we keep getting the high sign. So I want to give everybody here a last word. I want to make sure everyone here got to say what they wanted to say about the commitment to ultra-rare, or if there was something, a tool or a resource you needed to better serve the small population of the rare disease community, and you had a magic wand, what would that be? So I want to give everyone a chance to speak to that.
Okay, well, looking at me, I would start with getting a overt statement, a clear statement of a pathway that takes into account the totality of the evidence rather than sort of pretends to rest on adequate and well-controlled clinical trials as a way to reach a pathway to the market. This type of approach might require, like accelerated approval, a slightly tighter, but with, say, ultra-rare diseases, you're going to want to follow every single person who gets that because, you just need to collect. You're never going to have as much evidence as you would like to have, and you'll still have a lot of residual uncertainty. So there would be, caveats associated with it, but I really think that would help the reviewers, it would help the industry and the investors on a clear pathway, and most importantly, it would benefit the patients.
That and I'm going to go a couple of different ways. One is just to say thank you for all of what you're doing because I think just the collective chorus and the voices within this room are incredibly important for helping us drive the development of the policies, the thinking and the science, and helping really push, I think, the need and value of what patient advocacy and patients mean to the research itself. So I think that we need to keep doing that and do that more. And then I guess the other thing I would say is to help us think through, perhaps how we can help you more. Reach out to us and let us know how we can help you more.
Yeah, no, I won't add a whole lot except to say that I think with more than half the products in our center, and it's actually now more than half the products in the Center for Drugs, I'm told, in the rare disease space. I think there's a deep and abiding commitment here to improving what we're doing in this space. I think continuing the dialogue is really important, and if I had to ask for one thing would be the engagement to try to help move the manufacturing, the production in these products to a different place than where we are now, because that will benefit not just rare disease patients, but it'll benefit society as a whole.
So I'm just going to close by saying thank you to all of you, and I'm going to extend that thank you to, I know in the audience we have Janet and Catherine and Julie somewhere, and if I'm missing anyone from a federal agency, I apologize. But I just want to say thank you for your service to our community and for all the work that you do, but I also hope you take that back to your teams as well. The evidence bears out in the individuals who are living fuller, longer lives. We've heard a lot today about the struggles in rare and ultra-rare, but we also know that we've had some real tremendous successes and that we have generations of patients in rare disease communities that were not alive previously who are now going to school, going to prom this month, crossing the stage at graduation, and planning their families. And what we're fighting for is so that all of us can share in those same joys and successes. But we know that what we're doing so far has been working, and that is evidence. And we thank you for what you are doing. Thank you.
Well, it's hard to top that. In fact, we won't even try. That was incredibly thrilling, a very stimulating discussion, and we thank our experts for being positive change agents. I'll take the opportunity to also recognize that we talked a lot about data sharing and the importance of enriching each other across communities. Dr. Woodcock raised the example of Critical Path Institute, which was co-founded in 2005 by her. And that it's one thing to say we ought to do this, it's another to follow through, and that institute now has almost 30 consortia which virtually touch almost all of the rare disease communities as well as prevalent. So that's really something special, and I want to recognize that. To just recap, there's no way to summarize what's been transpiring and all the really great comments, the contributions of all the panelists. I think that's been a tremendous body of evidence that suggests there is a sense for change here, that there seems to be at least a general agreement that some kind of definition of ultra-rare is needed, if for no other reason than to provide a community focus so that other things can be built around that. So that, at minimum, would seem to make the most sense. So if indeed we proceed along that pathway, then, well, first of all, there will be a report of these proceedings, so this isn't the last step, and it's an opportunity to reflect on it. But how do we move ahead from this point so that the momentum is not lost? If there is to be a designation, what's the criteria? Is it numerical? Is it numerical in severity, as we talked about, or is it more operational? So defining a particular number this afternoon is not the goal of this enterprise, but to help folks think through how can we distinguish a population that can then be advantaged by appropriate incentives and other steps to improve research focus and beyond. So what is that kind of tail on the ultra-rare kite that we would all hope for? There would be visibility, and then that would be important for our communities to know that someone cares for them. that first statement, hope is informed optimism, that we saw is pretty compelling. So how do we catalyze that? Second would be the research focus already mentioned. Third would be the collaborative focus, that once people have something they feel they belong to, they're attracted to it. They commit time to it, they help each other, and that would be what we would hope for. And then we had these incredible additional suggestions that we've heard from Frank and Janet especially about some kind of comprehensive rethink on pathways, on benefit risk, on really looking at totality of evidence as opposed to strictly defined data. That doesn't mean we throw away p-values or substantial evidence. It means that that would be the ultimate form of regulatory flexibility that we've been talking about. So we also saw an example of another option, that being conditional approval, that is something that has been used in Europe. It does have its challenges, but it's another kind of pathway to look at as distinctive that may satisfy the rare disease community. We also had some other great things that probably connect to the research focus, or others like simulated controls, the data sharing, the linkage of planning to screening, and rare aware, if you will, to detect earlier. So without belaboring points, I think that there seems to be a sense of common understanding that ultra-rare would benefit from having its own day in the sunshine, and that appropriate incentives and so forth may be added that would allow us to look at, if not formal legislation, which is far and away from where we are, but the kind of things that were alluded to with regulations or other ways that could enhance regulatory flexibility. So those were the main points that were captured here. I've got a lot of other notes, and I know Jillian and others are going to be assisting us in summarizing. But I would offer those thoughts as this has been a tremendously energizing meeting. When you look at the unmet needs, the opportunity to address them, and having all the right principles in the same room to express agreement and support. So we realize there are certainly points that need to be worked out on these things, but the general spirit of the room, at least as I detect it, is that this is move ahead, be thoughtful, engage our communities, and figure out how to make this distinctive and advantageous for the patient groups being covered. So without further ado, Annie, do you have any additional comments? And if not, give yourselves a round of applause, because the participation has been fantastic. And with that, safe travels home, everyone. All the best.